Hello, is my voice going?
Yes, sir.
Am I audible?
Yes, sir.
Yeah, but I cannot hear your voice.
How about
Asha, ma'am?
now?
No,
Hello,
Dr. Anus, I cannot hear you.
sir. Am I audible, sir? Dr. Archer this
Asa,
side.
no, I cannot hear you too.
Okay.
Go to your audio settings and change your audio
Is there
settings.
something wrong? My
Ma'am,
voice is
voice
going,
not
but
coming.
I'm not able to hear anyone's voice.
Well, actually, you are audible to everyone, but maybe you're not audible.
Manish, sir,
Is speaker?
confirm for me.
Yes, ma'am, you are audible.
$37.
Sir, can you hear us?
Sir, am I audible? Please confirm me.
I'm Dr. Naresh. He is not able to listen us.
Dr. Naresh, just a minute, just a minute.
नहीं उसकी आवाज अपन को आ रही है
Yeah, hello?
Yes, sir.
Yeah, same thing.
Dr. Naresh, we
I'm
are
not
audible.
able to hear.
Sir, you are audible. Am I, sir? Sir, anyone from outside, please confirm. Am I audible?
Yes, we can
Atul,
hear
sir.
you.
Hello?
Okay. Good morning, one and all present here. I, Dr. Neha Sharma, Associate Professor, Organizing Secretary of International Conference in BioNano 2024. Welcome you all in session 4. This session is mainly based on animal and medical biotechnology and ethics in research. Today's chairperson is Dr. Naresh C. Bal, Associate Professor, KIIT University, Bhubaneswar, India, and co-chairperson is Professor Dr. Vinay Dwivedi, Director, Amity Institute of Biotechnology, Amity University, Madhya Pradesh. Now it is my responsibility to introduce our first speaker, Dr. Naresh Chand Bal. He is Associate Professor, School of Biotechnology, KIIT University, Bhubaneswar. He has more than 15 years of research experience in muscles and bone adipose tissue thermogenesis using various type of model organism. He is expert in development of mouse model, transgenic, pharmacological, surgical, and diet intervention. He is regarded as the discoverer of physiological relevance of small peptides called sarcolipid expressed specifically in the straight muscles. He has authored more than 50 manuscripts on this topic, including top-tier journals such as Nature Medicine, Trends in Endocrinology and Metabolism, Biological Review, Journal of Biological Chemistry, and Journal of Endocrinology. He has been awarded with several grants and honor, including Physiological Genomic Fellowship from the American Physiology Society, USA, award from National Institute of Health, USA, Ramalingan Swamy Re-Entry Fellowship, Government of India, Early Career Research Award, Government of India. He served as associate editor in journals like Frontier in Physiology, Metabolites, and Molecular Medicines Reports. Once again, I welcome you, sir. Please deliver your talk. Please, sir. Sir, please unmute yourself.
Okay. Hope I am audible. Okay. Thank you. Thank you. And I first thank the organizer for giving me this chance. And I am very uh, ashamed that this is a very uncomfortable situation. I can not hear you guys, but you can hear me. I'll try to address any concern that you have. If you put message on the text box, I can see them and I can address. So as the organizers told me, I have to complete my talk within 20 minutes. So I'll try my best. And the uh, deaf man's role that I'm playing today, I'll try to make it as much as amusing for you. So the topic that I have chosen is very close to my heart. I am choosing this skeletal muscle. Everyone knows skeletal muscle when it comes to the meat. Many meat lovers would be there in the audience. They would definitely know about what is skeletal muscle. Even those who do regular physical activity, exercise or yoga, so they would know what skeletal muscle is. But I am taking this as a target to counter metabolic diseases. That's what I have been trying to do last 10 to 12 years. So whenever you go to a doctor with diabetes or obesity, the first suggestion the doctor would have is do increase your physical activity, do exercise. And many of the literature in the scientific world tells you that if 30 minutes physical activity can reduce the chances of developing type 2 diabetes by 40%. But why? That is still very not fully conceived. So in US, the obesity is really a big problem, and about 1980s to 2010, there has been a huge increase in people with obesity in US, and India is nowhere falling behind, and we are kind of chasing this trend. In 1990s, there has been very low abundance or low appearance of obesity, both in case of men and women, but it has increased a lot within last 20-30 years. So the problem of obesity is huge, and uh, mainly because it increases the health cost, and the it worsens metabolic condition in several of the diseases, even including COVID-19, that we have learned very recently. So, in a nutshell, the obesity has been considered as a problem in energy homeostasis when our energy consumption or food intake is higher, and the use of energy is low in the body. Then there is a balance uh, energy which is not utilized, and that energy is stored as fat. So in a nutshell, it has been proposed as that. And recently, not recently, but yeah, close to 2 decades ago, many researchers started to propose an idea that we can increase energy expenditure, and we can dissipate the energy so that there would be less energy for storage, and when there is lesser energy for storage, the fat depot would be less, and if the fat depot is less, the associated problems can be reduced. But how to do that? How to increase energy expenditure has been a daunting task. The first one is using physical activity or using skeletal muscle based contraction, and do exercise. That's why the first doctors advise that to increase your physical activity. But something is there in our body what is called as non-severing thermogenesis. We are endotherms or we are homeotherms. We maintain a constant body temperature regardless of the fluctuation in ambient temperature. So not all the thermogenic demand is coming from only physical activity. There is something called as non-severing thermogenesis. So the site where non-severing thermogenesis occur, probably that is the target of increasing energy expenditure. That has been thought process. So where the thermogenesis occurs as non-severing thermogenesis, the organ has been characterized, that is called as brown adipose tissue. Brown adipose tissue is in human is usually found in case of um, uh, young children and newborns. But as we progress towards teenagers, the abundance of brown adipose tissue is lost. So the other site of non-severing thermogenesis, my research has shown that muscle. So before we go to muscle, just to briefly capture that the brown adipose tissue are mitochondrial laden adipose tissue that use a protein called UCP1 and lead to proton dissipation and convert the substrate into energy of heat. But in many organisms in the nature, they do not have brown adipose tissue. One of those is pigs, but still they are mammals and maintain a body temperature same as humans. And even birds, they do not have brown adipose tissue at all. UCP1 is mutated in them, it's not functional, and they maintain a body temperature much higher than mammals. So all those animals indicate that there is something else which also has non-severing thermogenesis. And studies in fishes has shown that skeletal muscle uh, can be converted into a heater organ. So based on that, I started working on the idea that how skeletal muscle metabolism can be tuned off to um, non-severing thermogenesis. So initially there is for how skeletal muscle works. Let's see that this is the calcium uh, uh, flux that works for contraction of the skeletal muscle. This is for cardiac muscle, but skeletal muscle also works in the same way. So the calcium during the contraction process starts to induce the contraction, and when the muscle has to relax, there is a protein called SERCA which has to suck up the calcium from the cytoplasm to the inside of the SR of the skeletal muscle so that the muscle can relax. So this circus function is regulated by two transmembrane proteins. One of those is sarcoplasmin, which is my main focus of research. So I have been working on sarcoplasmin, a small transmembrane peptide, and I have shown that this protein, when knocked out, it leads to thermogenic imbalance. And two back-to-back nature paper supported my idea, and that's how we established that sarcoplasmin is a protein that is involved in uh, non-severing thermogenesis. So for that, we also developed a surgery that is the brown adipose tissue. Just I was talking about brown adipose tissue. This is the brown adipose tissue located in the outer side or dorsal side of the mouse, and we have taken out the major chunk of brown adipose tissue and forced them to cold challenge and various pharmacological testings, based on which we have demonstrated that sarcoplasmin is a um, kind of tweaker of the circa activity, and through which it contributes to uh, thermogenesis. And in normal animals, when they are fed with high energy containing diets, the sarcoplasmin is super induced in some of their muscles. That kind of protects them from that kind of high fat diet uh, side effects. So based on that, I got several questions, and I have been working on those questions. How sarcoplasmin is intertwined with muscle metabolism? The first thing that we found out is the mitochondrial biogenesis that induced by sarcoplasmin. So sarcoplasmin overexpression mice, the muscle becomes hugely flooded with amount of mitochondria. And in another study, we have found out that UCP1 knockout, UCP1 is the protein that leads to heat dissipation in the brown fat, and that protein, when knocked out, it induces sarcoplasmin. This also indirectly pointed out that skeletal muscle plays an important role in non-severing thermogenesis, and sarcoplasmin is involved in that. So with these studies, I moved back to India, and in India, I have been studying about how this sarcoplasmin is intertwined with metabolic disease. And I have found out that the skeletal muscle and the brown fat, they have lot of intricacies among themselves. They functionally in kind of crosstalk to each other, and when one is reduced, the other one comes to compensate. So these studies have taken a lot of interest in the field, and we are getting a lot of follow-up studies on this. And we have been trying to understand how the skeletal muscles lack of sarcopene or increase in sarcopene expression help in a diabetic situation. So one another study is under process, and we have some data to show that the sarcopene help in glucose clearance of the body. So one of the compound that I have come across from literature is capsaicin. Capsaicin is a compound that is found in the red chili, and that capsaicin helps in stabilizing the interaction between sarcopene and sarca. We have some computational studies and some biochemical studies to show that when sarcopene is there, the sarca and sarcopene interaction is stabilized. And now we also have tissue level studies from sarcopene overexpression mice to show that capsaicin can increase the or enhance the calcium uh, sarca's ATP utilization capacity. So based on this, I am going towards understanding how capsaicin can induce muscle energy expenditure. And we have been designing a lot of different analogs. And today I chose not to show a lot of more data that we have currently developed, how capsaicin can increase energy expenditure due to shortage of time. But the intricacy between the brown fat and the skeletal muscle is another facet of the same coin that I am going to study. And another study is almost there, in which we have found out that the brown adipose tissue and skeletal muscle metabolism is intertwined when during development in a human-like model using goat as a model system. Another study, we are using bird as a model system to study how skeletal muscle based metabolism is able to perform whole body energy expenditure. And to conclude my talk today, the sarcopene is a recruiter of pathophysiological conditions in the muscle metabolism, and sarcopene's role can be enhanced by pharmacological means to increase energy expenditure to counter obesity and type 2 diabetes. So this is my group, and these are my collaborators. This is my group in KIT, Kalinga School of Industrial Technology. So we are located in Bhubaneswar, and Bhubaneswar is also called as a city of temple. I request all of you to find some time to visit us in Bhubaneswar. We'll be glad to host you. Thank you. Hope I did not exceed my time.
No, sir. You will finish within the time, sir. Thank you so much.
I cannot hear you.
Right. Thank you very much, sir. Well discussed topic and interesting topic. It will be surely benefit for us and our students. Thank you once again, sir.
Dr.
We
Naresh, I
will
think
take.
am I audible?
No, sir. No one is audible. Today.
Yeah, I can hear from your mobile. Yes.
We will take all question and answer in panel discussion at the end of the
Dr.
session.
Naresh, this is Dr. Vinay Dwivedi.
Thank you, sir, for inviting me. Yeah.
Okay. Actually, one of my colleague, Dr. Snehashish Mishra, he is with me during my tenure at IMS Engineering College. I think he is professor there.
Yeah, yeah, yeah. He's
Yeah.
a great colleague here.
Yeah. He was my colleague in IMS Engineering College. We both were working there.
Okay, okay.
And
Okay.
I visited KIIT, I think, in 2013.
Oh, I see. Long
When there
back.
is international conference in biotechnology department, at that time I met with Dr. Snehashish and Dr. Samantha also.
Oh, I see. I see. I see.
Yeah.
I see.
And
Okay, sir.
Dr. Mrityunjay Suar is there?
He's there. He's no more the director of our department, but
Okay.
he's DG Research and Innovation in
Okay,
KIT.
okay, okay.
Yeah.
Okay, okay. Nice to hear you, sir. Thank you.
Okay.
1 second.
एक मिनट.
Hello, Dr. Naresh.
Yes.
Please unshare your screen.
Yeah, yeah, yeah. Okay.
Thank you once again. Now the next speaker is Dr. Anuj Sharma. Dr. Sharma is virologist with expertise in the field of arbovirus pathogenesis and vaccine development. Prior to joining the Office of Research Integrity, he was an assistant professor at Uniform Service University of Health Science, Bethesda, MD and the senior scientist at Henry M. Jackson Foundation. Dr. Sharma completed his PhD from the Birla Institute of Technology and Science, Pilani, followed by postdoctoral training at USUHS. He is recipient of grant from National Institute of Health, Department of the Defense Radiation Health Program, and Defense Threat Reduction Agency. In addition to virology, his research interests include radiation countermeasures and traumatic brain injury. He has published over 30 peer-reviewed papers and 4 book chapters. Once again, I welcome you, sir. Kindly start your talk.
Thank you, Dr. Sharma, for a kind introduction. Thank you to the International BioNano 2024 for the invite, and I'm happy to be here. Now, although I am a virologist, I still consider myself a virologist. I moved from the full-time research to a different kind of work that we do related to science, which is research, looking at research misconduct in research. Specifically, so I want to discuss today a little bit, pivot away from our regular hardcore science work to a little bit more of a different side of the science, that if when a research misconduct occurs in the research, how it affects science and how it affects researchers. And of course, all this is an angle from what I have learned at ORI. Before I go further, just want to give a disclosure that these are my opinions and does not reflect that of the Office of Research Integrity or the Office of Assistant Secretary of Health. So I hope that this talk is interesting to everyone who is listening, including the students. My aim is that, when we move away from this session, that the research integrity and ethics and every other aspects of moral integrity that we have in conducting science is always in the back of our mind. The U.S. Office of Research Integrity, organization-wise, is under the Office of Secretary, and there are 2 different offices or branches within the OS, which is one is Office of Assistant Secretary of Health, and the other are the operating divisions. So there is a kind of separation between our office, because we are the watchdogs of scientific research and any allegations that occur in research misconduct. And therefore, we have jurisdiction over all the public health service funding agency. Most common that everybody knows is National Institutes of Health, or NIH. We also have regulatory or oversight authority over CDC, FDA, some part of the FDA research, and many other agencies that give out funding for scientific research, either internationally, nationally, intramurally, or extramurally. With that, all of those research is under our purview. So our office was established in 1992 by the act of the US Congress. And the mission is to protect the integrity of the research, specifically of the research that is funded by US taxpayer dollars, specifically for public health service funding. And also to indulge in promoting research integrity. And that's why you find me here in this session. All of our work is governed by Code of Federal Regulations. So that you can consider that as our Gita or Bible or Quran, whatever you follow. But essentially, that's what we follow, the Code of Federal Regulations, while dealing with allegations of research misconduct. So within the Office of Research Integrity, there are two different divisions: Division of Investigative Oversight, of which I am a part, and the Division of Education and Integrity. As the name suggests, you can imagine that the DIO deals with the allegations of research misconduct. They also deal with monitoring research misconduct proceedings that occurs at institutions. Finally, we have an oversight authority. So we do an oversight review of all the research misconduct proceedings that occurs at institutions. And if needed, we make a recommendation for PHS, separate PHS finding. That means that our finding is made public and it is separate from that of whatever the institutions may have found or may have made a determination of. From the DEI side, they deal with assurances. So how it works is that any institution in the United States or any institution that wants a USPHS funding for their research, they have to file an assurance with our office, which says that they have policies and procedures in place that if there is an allegation of research misconduct, they will deal with it and address it in a compliant manner. So DEI side deals with the assurances, compliance with the assurances, outreach programs, and etc. So how do we actually look at an allegations? When we receive an allegation, of course, we do an assessment, quick assessment by ourselves within our office. And if we find it that it is reasonably okay allegation or believable allegation, we forward it to the institutions where the work was performed. And then the institution has 30 days to finish their assessment, file a report with our office, saying that what they have found in the assessment, if they are proceeding to an inquiry. For an inquiry, an institution gets 60 days. That reports get filed back to us. We do an oversight review to see if everything is in compliance or not. And then if the investigation is warranted, then the investigation has 120 days to finish. The final report is submitted back to us, and then we do an oversight review, look at all the evidences, look at if the findings are based on the evidence that at hand, and then we can take administrative actions. And some of our administrative actions that we recommend to further up the chain can be debarment. That means that we found that a person should not continue in scientific research and should therefore not participate in any PHS funding matter. So either it be performing the research itself, being a consultant, advisor, reviewer, nothing is allowed under debarment. Then we have just other things which, they are allowed to do the research, but their research has to be supervised, or the research has to be certified by a committee of peers and the people who are expert in that area of research. And we can recommend retraction of publications as well. So what is research misconduct? That's the main part, right? Many people can come up with any allegation saying that, hey, we think that this is wrong, this is research misconduct. But for us, for ORI, the definition of research misconduct is falsification, fabrication, and or plagiarism. And the next word is really important, proposing. Proposing means that even if you have falsified data included in a grant application, then that is considered research misconduct, even though it has not been funded yet, but still we have jurisdiction over there. So in proposing, performing, or reviewing research, or in reporting the research results. Fabrication is making up of the data. It makes sense. Falsification is when the data is misrepresented. So suppose somebody got a negative results in their experiments, they kind of fabricated the things, reported the experiment to be working, then there is falsification and fabrication. And plagiarism is something that we are more familiar of, that you take somebody's ideas, words, text, and claim it to be yours and don't give the person or the resource appropriate credit. What is not research misconduct, especially for us, and most of the things under this list is usually taken care by the institutions. It doesn't mean that there is no misconduct. It's just not research misconduct for us. And that is honest error. Like somebody had the data but made a mistake. So that is acceptable. Authorship, collaborators, disputes, duplicate publications, misuse of animals, or protocol violations, conflict of interest, all these are not under our jurisdiction, and we do not consider that as a research misconduct for ORI point of view. So how do you make a finding of research misconduct? The research misconduct finding have three main requirements. One is whatever happened should be a significant departure from accepted practices. And the committee should, and it has to be committed intentionally, knowingly, or recklessly. And finally, the act of research misconduct has to be proven by preponderance of evidence. So, on the left side, I have a triangle for you, which says perceived pressure, rationalization, and opportunities. And I have adopted it from Creasy's fraud triangle. What it says is that why people commit research misconduct. There is perceived pressure. Hey, I need to publish. I need to get this paper out. I need to get my PhD. I need to, get to my other job. Whatever it is, there is no real pressure, but people perceive a situation as a pressure situation. Rationalization. Sometime researchers may think, hey, it's some really minor thing. Nobody is really going to bother about it, or it's not significant. It doesn't matter. I just need to put this in because somebody wants it to be there. Or an opportunity, which opportunity is also a really critical factor, and we see it all the time. When what happens is there is no checks and balances in place, and lack of checks and balances, verification of the data, provenance of the data, management of the data, all that leads to an opportunity for research misconduct to occur. So, if you're running a lab, if you are interested, if you are involved in research misconduct, I think the experiment is important, but how you preserve the data, how you process the data, how you validate the data is equally important. Before you put it out for public consumption. So I want to give you three different examples of the cases in which ORI made a public finding. And my idea here is to show how it affected the science and then how it affected the researcher who committed the research misconduct, just trying to tie up with the title of my talk. So in this case, this was a senior scientist with more than 10 years of flow cytometry experience. She was listed as an expert in flow cytometry at many places. But when the principal investigator was writing the paper, he saw that the data was not really making sense. Essentially, the raw data from the experiment did not really match the final data that was presented to him or her. So the science here in question is a vaccine platform development for HIV, specifically using the HIV-1 gag protein. What happened here was that there were a bunch of flow cytometry experiments, and looks like everybody is out. A bunch of flow cytometry experiments, which resulted in falsified data included in more than 10 NIH applications. And what happened here was, this is the data looks like. What the researchers showed was that when the vaccine platform was used, it resulted in a robust interferon response, showing a nice CD4 memory T cell response in these experiments. However, when the data was looked closely, they found that the gating was different. So for people who don't understand flow cytometry, gating is a very important and a very basic requirement for flow cytometry. You set up a gate in which is then uniformly applied across all your samples to get the comparable results. However, in this case, what they did was, for the non-stimulated samples, it was gated on almost the cell debris, and for the stimulated samples, it was gated at where it should be. And what they were trying to show is that upon stimulation, you have a nice CD4 cell response. But what the real data actually showed was, when the gating is done correctly, that there was not really any difference. So by changing the gate for the samples that they wanted to look at or wanted to show some response, they departed from accepted research practices. That result in falsification of the data, because the data does not show CD4 response. And then that shows the fabrication of the results that the whole platform is working. So falsification and fabrication occurs here. ORI made of public finding of research misconduct. You can find it under the federal notice. I have the citation here. But essentially, this person was then suspended for 3 years. Not really suspended, has a requirement of 3 years supervision. So that means wherever this person works, there has to be a committee of expert who oversees her work, checks that she has actually performed the work, and then that work is certified that this work is actually true and reflects the real results. And then this person cannot serve as a reviewer or on any of the PHS matter. Scope here was big. It was falsified in 10 different grant applications. More than 20 figures were affected. So, you see that there are the effect on the science is that whatever was proposed to be done new from here was based on a falsified proposition. And that resulted in another study, which I have not talked here, but another study which wasted the whole, higher animal model efforts. So a lot of wasted resource, a lot of wasted, animal model efforts, and as well as the time and money when the research misconduct occurs. In the second example, this person was a clinical research coordinator. Again, the raw data on the instrumentation did not match the final process data. This was more on asthma and COPD, essentially looking at environmental irritants which causes or exacerbate the COPD symptoms. And in this case, this person fabricated all the numerical values to show that their specific lung function measurements were working and responding to what they were looking at. Scope here was big. There almost 94 PHS grant funding were involved. So almost 100 grants resources were wasted. The whole point is that whoever read those manuscripts and the research records and the paper, there were 39 papers that were affected by the falsified data. So huge effect. And I wanted to show you this is not just limited here. So we made a finding, this person cannot ever work on a scientific project that relates PHS funding. It's just not allowed anymore. There was Department of Justice got involved, and there was the institution where the work was done was found to be careless, and that institution was fined $112 million. And there was the state where the institution is also made a finding of an embezzlement of funds, and they put basically more fine on the institutions. So here, everybody was a loser because of one person who was not doing their job perfectly, and so effect of this on the science was huge. Effect on the person is huge. Effect on the institution was huge in this case. A third and the last example I want to show here. This person was an associate professor, so pretty senior in the field. And again, whatever was looked at was all falsified. All kind of data was falsified. I just want to show you two. The science in this question was cancer stem cells, essentially showing the drug sensitivity to for eliminating breast cancers. So the experimental model was in question. Key molecules that were identified in this therapeutic targets were all questioned. Just want to show you a snapshot here that one single blot, the two bands were used to represent 4 different proteins here. And you can see that they have all been presented in a different way, clearly showing that the person was it was an intentional act. If you do a little bit more analysis, you can see on the right side in the blue images that the first 2 lanes are kind of same. Confocal microscopy, they use the same data to represent results in 2 different models. One was Homo sapiens cell lines, which is MCF12A, and another was mouse memory stem cells. Departure from accepted research practices. Of course, you say it might have been the same results, but you cannot use a memory cell, a mouse cell data to represent a human cell data. That's a departure from accepted research practices, resulted in fabrication of the data and falsification of whatever their finding was. So kind of like, destroying the whole research there. In this case, there is a 10-year debarment on this person. So for 10 years, they cannot work on any PHS grant, period, anywhere in the world. You cannot just leave US and go and work in anywhere other country and be involved in the PHS funding. You cannot do that. For 10 years, they cannot serve as an advisor or a consultant or a reviewer or for any PHS matter. The papers were reflected. And just to give you a snapshot of, there were about 384 different figure panels were falsified. So huge impact on the science itself. Think about all those papers that were taken by people and then cited and believing that this research was true, and then did their own research, and probably half of the time would have been figuring out why their results was not working. So the impact of any falsification, large or small, is huge on the science. And you are seeing here that its impact is huge on the researcher. You come into research as a passionate researcher, right? And you go through all the grind of the postdoctoral fellowship, the assistant professorship, associate professorship. And then if you find that there is a falsified data, then you lose all those years of your own effort for what? Just for a few papers or whatever the reason, people find themselves to be committing research misconduct. So last 2 slides, the take-home message for what I want to give to almost everybody in the audience is the impact of research misconduct is far-reaching. If it is unreported, unfound, then that percolates in the literature, in the scientific field. For example, this is a little bit older study, but there were 164 publications, were followed. They had falsified material in it. They were cited 7,000 times, about 7,000 times. So that is direct, basing on the falsification containing papers. And those 7,000 were cited 301,000 times. So 3 lakh more publications were based on that. So that's what I mean when I say that the research misconduct percolates in the scientific literature. You saw what happens on the effect on the researchers. They lose their career. Many times they lose their career in scientific field. So one thing I want to say is that, Mark Twain, he said that if you tell the truth, you don't have to remember anything. But not really. I kind of disagree with him. In a scientific research, you need to maintain your records. You have to have a good data management. You cannot say that I know that it is truth, and that's why you should believe it too. You should be able to prove your truth. And that in science can only happen when you preserve your data, when you have checks and balances of verifying what you are publishing, what you are reporting. And that's why it's hugely important to not forget that you should maintain a provenance of your data and the validity of your data. And I would say that, report research misconduct. I'm not so sure how it works in India, but, if you see something wrong, then you say it out and you point it out, and that goes far. So you can find more about ORI on our website. We regularly publishes our case summaries if you are interested. I know not a really passionate subject to go after, but still you can find that out there. I would like to acknowledge my colleagues, Sheila Garrity, she is our director. Alex is our director of the division which I work in. And some of the case examples I have shown you is from my colleagues as well, and for me and my colleagues as well. So, a big thanks to all of them. And then I will end. Thank you, and I can take questions at the end.
Thank you so much, sir, for very informative talk. I have found personally that every talk is very interesting and we need more time to discuss it. And sir, it's true that many researchers lost their job and they have to maintain their data for continue. Thank you once again, sir. We will take all question and answer in panel discussion, sir.
Thank you.
Now, our next speaker is Dr. Atul Sharma, Assistant Professor in Department of Critical Care and Respiratory Care, Division of Anesthesiology, Critical Care and Pain Medicine, University of Texas, MD Anderson Cancer Center. He did his PhD in molecular medicine in 2012 from Jawaharlal Nehru University, New Delhi. He has started his journey as Junior Research Fellow from JNU, New Delhi, after that SRF, and did postdoctoral research from UND School of Medicine, Grand Forks, ND, USA. He has published 17 research papers in different international and national journals. Once again, I welcome you, sir. Kindly deliver your talk, sir. Please, sir.
Thank you for a nice introduction, and I'm very happy to be here. Just let me quickly share my. Okay. Yeah, I hope it's visible clearly here.
Yes,
Okay.
sir, it is visible, sir.
Thank you. So I'm going to talk about the neutrophil extracellular trap, which we are mainly I will be calling them NETs. What's the mechanism of net formation and the therapeutic intervention using them on at modulation. So as we, I think most of us know that neutrophils are the primary respondents upon any bacterial infection, and they are the one first to reach at the site of infection in the bloodstream, through the bloodstream, and they fight with the infection. Initially, it was thought that they are doing it by eating away the through the phagocytosis, eating away the microorganisms and controlling their growth and or the pathogenesis indirectly. And later on, in 2004, it was discovered that they release mesh-like structures composed of DNA fibers, which act as a, and that's why they call it NAV, which acts as a trap to immobilize the pathogens, and they have a lot of antimicrobial proteins and enzymes which acts in elimination or killing of the microbes, the trapped microorganisms. So relatively, they were discovered much later or recently, and then thereafter there have been so many new researchers have come. So the primary goal, and this was published that time also, it was the primary goal was how to just protect the host from invading microorganisms. And like I said, through they're decorated with the antimicrobial proteins. And then slowly there emerges another side of these nets that way, if you think that way, the nets are good. I mean, the bacteria comes in, the neutrophils go there, release their net fibers and kill them. So it's good to have nets. But slowly it was not that simple, and that's the whole thing, the whole fascination about this topic, because they found that if you have a lot of nets, if you excessive net, it results into a lot of other diseases, including all the autoimmunity, including the acute lung infection, arthritis, lupus, Alzheimer's. So because of the autoantigens which are present on these excessive nets, they derive the autoimmune response, resulting into many disorders. So now we know that, or at that time we knew that the nets have protective as well as the pathological functions, and that highlights the need to understand it and to if the mechanism through finding other mechanism, as well as if we can modulate these nets so that to manage their disease conditions. So and like I said, it was discovered pretty late. So that time the main process was, forget about the mechanism, was what is this process of net formation, or we start calling it netosis. That means initially they were found the microorganism, there are certain cytokines, and there are other molecules they found initially, and which triggers, but basically the mechanistically they found that the ROS, reactive oxygen species, that triggers or that initiates the transformation of neutrophil to a netting neutrophil. So that's why I call it netting neutrophil. And since that time in 2012, when we were quite fascinated because we were in an infection biology lab, and we were quite fascinated. So we also started our own approach or our own attempts to find out the mechanisms as well as understand this phenomena better. So these are the images we initially found when we were able to stimulate the nets in vitro using certain chemical reagents, mainly PMA, which I will be talking about more in the coming slides. These are the first view of the nets which I was able to see and when we really started. And then what we found interestingly that because our was infection model where we used to infect Klebsiella pneumoniae in the lungs of mice, as well as the genetically knockout mice of Minkel, which is a macrophage-inducible calcium-dependent lactin. We found interestingly that upon infection, the lung of wild-type mice shows the structure which we confirmed using the neutrophil elastase antibody, which is a marker of NAT, as these are confirmed NATs, but these structures are absent, and these mice were super susceptible. And if you see the pathology, lung pathology, there are huge influx of neutrophils, and so then we thought what was going on. There are a lot of neutrophils coming in, but these neutrophils are not able to give protection, and the mice are very susceptible. They are dying. Second day of infection, they are showing high clinical score, and they are really, really sick compared to wild-type mice, which were able to resolve the infection after 5 days. So then we thought, oh, they are able to clear the infection because they have nets, but there are no nets. So this was pretty interesting, and that's what we published initially. And of course, we took it forward, and then, like I said, the initial trigger was considered as a ROS. But interestingly, in these knockout neutrophils or the knockout animal neutrophils, the ROS production was considered as normal. As you can see in the wild type and Minkel knockout mice, as well as we also did the siRNA, Minkel siRNA transfection, and that when we silence the Minkel gene, that time also you can see actually with PMA, which is a ROS inducer, there is no dearth of ROS production. And similarly, this is cytosolic ROS. We also found a mitochondrial ROS that Minkel knockout mice are not showing any deficiency in ROS production. So we thought then if the ROS is same, then why they are not producing nets? So we looked at another aspect, and we found in vitro first that the autophagy is defective. So the autophagy, which you can see, these are the punctas which marks the process of undergoing autophagy. These are the neutrophils upon stimulation with PMA. They are showing the LC3 puncta. LC3 is a marker of autophagy activation, but these are absent in the Minkel knockout neutrophils. That means the autophagy is probably, it could be a factor. And that's what we found in vivo then, that when we infected these mice, and then, like I said, they are not showing nets. But, and these are, you can see the net percentage. The ROS is same, but autophagy is defected in vivo. So you can see the absence of LC3 puncta. These cells are from the lung wash of these infected animals. And also you can see this is a Western blot. This is showing that all the autophagy markers are down in the mingle knockout, but they are pretty high or activated in the wild type mice. So then what we thought that how about we somehow increase autophagy through some reagent and see if that is down, and if we are able to through some drug which we can show the recovery. And that's what we used tamoxifen. That's when we used tamoxifen, which is a non-autophagy inducer. And we found that if when tamoxifen is present, the net deficiency is rescued. And even these mice, which these knockout neutrophils, which are not showing any nets, upon stimulation in vitro, they are showing that is rescued and that is recovered. So that was really fascinating to see that how a particular and then very interesting thing which I will be talking about more about, it does not increase ROS. It does not interfere with the ROS. It is purely working through autophagy. So then we thought, oh, we can probably through this we can let's see in the if we can do any disease pathogenesis recovery. And I'm not going to show there's some data we are still unpublished, but I can share with you safely that yes, we did find that with the tamoxifen treatment, we are able to rescue the survival, and we are able to really do better on the pathogenesis of these infected mice. That means through autophagy, it can produce more nets, and then through that we can help these mice recover from the disease. So this also we published later on after about 2014 paper in JID, and so this basically suggesting that this pathway is segregated from ROS generation, and then that's why through autophagy we can get the capacity of modulating nets. And then we thought that probably we may able to get this capacity of playing with this in other diseases where the net formation is either high or low. And if we can, so we picked up many disease models, COPD, neonatal sepsis, CGD, which I will be talking more about, and also sepsis. Since we covered a lot of these aspects, and it's due to time limitation, I won't be able to cover all of them, and some of them the research is still going on. I will be talking more about CGD because that's what we publish later on. CGD is a chronic granulomatous disease, and it is basically caused by mutation in a gene code in NADPH oxidase subunits. Because of that, these phagocytes are not able to produce ROS, and that's why they are very susceptible to all the bacterial and fungal infection. And neutrophil chemotaxis and phagocytosis are normal, but individuals with CGD are able to mobilize phagocytes to the site of infection, but microbial killing is impaired. So they have very high rate of bacterial growth once the infection is initiated. So that's why these people are always susceptible to any bacterial infection, and they are always under antibiotic and under treatment. So because there's autophagy defects detected in CGD, and so what we did that we take these CGD neutrophils from the patient blood and treat it with LPS, and we see that a healthy individual is able to make nets, but the CGD individuals are not able to make the nets out of LPS stimulation, which is a biological. PMA is not a biological net inducer, but that's why we use LPS. But when and you will see some of the places RX1, which that time we were using because we didn't, but now this is a tamoxifen, which is another name of tamoxifen we were using at that time. And we saw that with that, we are able to recover the net deficiency upon LPS and PMS stimulation, as well as on bacterial. So you can see that a healthy individual neutrophils, when you put the bacteria in, it makes a lot of nets, these nets. But when you, in case of CGD patient neutrophils, they are not able to. But when you feed them with the tamoxifen, they are able to make a lot of nets. So that way, you can see that they are able to control the bacterial proliferation. And interestingly, similarly, we found that upon tamoxifen treatment, there is a generation of our LC3 puncta, and that we found again that is playing a role. And when we use NSC, which is a LC3 inhibitor, specific LC3 inhibitor, we see that these punctas are down. Again, that resulted into a publication in Journal of Allergy and Clinical Immunology, where we showed that tamoxifen works in absence of NADPH oxidase ROS. It does not, in CGD patient, is able to stimulate functional nets and is able to rescue the net formation and net-mediated killing against bacteria. So now, with that, like I said, our research broadens into therapeutic modulation of net formation, and where the nets are underproduced, for example, in CGD and neonates, or the neonatal preterm and term babies, as well as overproduction of nets, where we have situations like sepsis or the breast cancer. We are working on all these different disease models, and I was only able to share a bit of that, which was of the time limitation, as well as there are certain things we are still about to publish. But with that, I will just keep it crisp, and I will just share the publications we were able to get, some of the showing all these net formation and net related mechanism. And with that, I think I would just like to thank all our collaborators and the grants which the lab got. Of course, my PI, Dr. Jyotika Sharma. She's the one who directed all the studies, as well as she's the one who got all the grants. And our collaborator, Dr. Holland, Dr. Jerbe at NIH. And with that, I think I would just like to conclude. Thank you very much for your attention, and I would happy to see the questions after the I think all the panels are done. Thank you so much, everyone.
Thank you so much, sir, for very smooth talk, which was very informative and have lots of knowledge for students and us, sir. Thank you once again, sir.
Thank you.
Now, our next speaker is Dr. Sai Manji. Dr. Sai Manji is a health scientist administrator at Maternal and Pediatric Infection Disease branch of the unique Kende Shriver National Institute of Child Health and Human Development. Her research portfolios focuses on pediatric immune system, ontogeny and development, immune prophylactics, therapeutics, and vaccines in HIV, TB, and malaria infections in infants, children, and maternal individuals. Regulatory affairs focusing on clinical trials, safety, monitoring, and pediatric COVID and MISC. Dr. Sai was with the Department of Defense for over 7 years, working as an immunologist on malaria vaccine development, developing monoclonal antibodies against malaria and HIV, testing candidate vaccines, and developing humanized animal models for malaria, HIV, and dengue viruses. She was also at the US Army Medical Research and Development Command as a senior regulatory scientist. I request you, ma'am, and welcome you, ma'am, once again. Kindly start your presentation. Please, ma'am.
Introduction, Dr. Sharma, and thank you so much to Dr. Mishra as well as Dr. Dwivedi for inviting me for this session. I'm going to try to share my screen. Please let me know if you can see my presentation. Let me make it full screen.
Yes, ma'am. It is.
Okay.
Please continue.
Thank you so much. So as per the introduction, my name is Sai Maji. I am a health scientist administrator. I work at the Maternal and Pediatric Infectious Disease branch at the Eunice Kennedy Shriver National Institute of Health, Child Health and Human Development. I wanted to give a very broad overview of what NIH is and go into a little bit more detail about introducing my institute, which is the Child Health Institute, and talk a little bit about what we do, both at the branch level, at the institute level, as well as share some tools and resources that might help you. So whenever you're thinking of applying for research funding, one of the biggest institutes that comes to mind is the National Institutes of Health. So the mission of NIH is to seek fundamental knowledge about nature and behavior of living systems, and the application of this knowledge to enhance health, lengthen life, and reduce the burden of illness and disability. So this is a very broad mission, and the way NIH accomplishes this mission is by supporting what we call as extramural research. So 80% of the funds that NIH disperses is to the research that is performed by investigators in over 2,500 universities, medical schools, and other research institutions, both in the United States as well as around the world. These funds facilitate training of new investigators, public availability of data, and also to communicate information via the National Library of Medicine. So the other 10% out of the 100 goes into intramural research. So there are actually research labs. I want to show that this is where the central building for National Institute of Health is. It's in Bethesda. There are multiple labs in Bethesda as well as other places in the country. And 10% of the support goes to fund these research labs. And the remaining 10% goes into the management of this research. So I am part of that 10%. So when we look at the organizational chart of National Institutes of Health, we are a huge organization of over 27 institutes and centers and offices. So the institutes are listed here. I don't know how many of you are familiar, but one of the speakers was being funded by the National Institute of Diabetes and Kidney Diseases, which is NIDDK. My institute is here, the Child Health and Human Development Institute. There are some centers like the Clinical Center, the Center for Scientific Review, and there are some offices like the Office of the Director. Each institute has a specific priority research area, and they perform research both intramurally and extramurally based off of the priority research area. So I wanted to talk a little bit about NICHT because it is a little bit special. Pictured here is Robert Kennedy, who was one of the US president, and that is his sister, Eunice Kennedy. Eunice Kennedy was extremely passionate about developmental disabilities in children. More importantly, at the time was Down syndrome and autism. So she really wanted a public institute, which was funded by people's dollars, to focus on research that basically looked at these developmental disabilities in children. She and her family are one of the biggest supporters of Special Olympics, and she firmly believed that everybody is created equal and need equal opportunities. So this is a very memorable picture where she is actually handing over the pen to President Kennedy to sign the authorization to establish NICHD in 1962. Just a little bit of history there, but our present, moving over to present, this is our current director, Dr. Diana Bianchi. As I said, our priorities are child health and human development, and our director is fully supportive of all our priorities. So a few years ago, one of the questions was, what is one of the mission or vision of the Institute? So we had to come up with something that describes the kind of work that we do. And NICHD's mission is to lead research and training to understand human development, improve reproductive health, and enhance lives of children and adolescents, as well as optimize abilities for all. Of course, that's a very big statement, but simply put, our vision is healthy pregnancies, healthy children, and healthy and optimal lives. So how do we accomplish something that is so broad? So we decided in 2020 that we were going to do a strategic plan. So in very simple terms, whenever we have a research or a proposal that comes to us for review, the proposal has to fall under one of these planned themes. So it has to be either to understand the molecular and structural basis of development. It has to be able to promote gynecologic, andrologic, and reproductive health. It has to be able to set foundation for healthy pregnancy and lifelong wellness, able to support child and adolescent health and transition into adulthood, as well as advance safe and effective therapeutics and devices for pregnant and lactating women, children, and people with disabilities. So this is, again, coming into cross-cutting topics, and these topics are integrated into all research themes. So one of the reasons I put this here is global health, which is health of the whole entire world population, is part of these research themes. And we are program officials, and as program officials, we consider ourselves scientific stewards. So we have the goals that are listed here as primary goals. So just to state a few, I'm not going to read the whole thing, but one of the things is to promote an inclusive scientific area, allow to set research priorities, and align resources in order to support this kind of research. And my favorite one is to facilitate transparency and communication. So as I described earlier, I belong to the maternal pediatric disease branch. I wanted to show my colleagues and people who I work with, we all have distinct areas of research. So Sonia is our branch chief. She works with, she's a psychologist by training and works in behavioral medicine. Franklin Yates is an MD physician who's a new recent person to join our branch. He focuses on adolescent HIV prevention and treatment, infectious disease and viral hepatitis, and so on. So each of us have an individual area of research. And when you submit an application to NICHD, it falls under one of our program areas. Just like the Institute, our branch has research priorities, and those priorities are listed here. And when we consider one of your application for funding, we have to align your research into one of these branch research priorities. Again, I'm not going to read these priorities. They are on the website for the branch, so they're open access. So one of the things I really wanted to highlight is how it's a maternal pediatric infectious disease branch. So we are, jokingly, we refer to ourselves as infectious. We try to look at infectious diseases all over the world. So we really have a huge global footprint. About 68 countries have received grant funding from NICHD. Of course, there's multiple countries in the African continent with a higher impact and more larger presence for the institute. But nevertheless, this is very impressive because we're among the few institutes that actually work very closely with Fogarty, which is the International Center, and fund multiple countries in the world. So one of the things I wanted to focus on in this talk is as a researcher, as an investigator, if you are thinking about applying for funding, how do you go about it? So one of the things that as a federal institute, National Institute of Health does is publish what are called as Notice of Funding Opportunity Announcements, or NOFOs. So whenever you're looking at a NOFO, one of the things that you would see would be contacts for a program officer or a scientific contact. There's a scientific review officer, and then there's a grants management specialist. So these are the three people as an investigator, PI, or your institute's authorized representative is usually in touch with. So the first thing you would do when you're in the process of inquiry or preparing for submitting an application is you find who is the program officer who aligns with your area of interest. And you speak to the person. You'll say, hey, this is the idea that I have, and this is what I'm thinking about. And you and your program officer have discussions, and say the program officer feels that this is an area of interest for the institute as well as priorities. They will immediately, have further conversations and allow allow you to apply as in their portfolio. So once you submit your application, it goes into review. And once it's reviewed, you receive a summary statement about how the review went. And program officers are available to discuss your summary statement. And once your application has been deemed fundable, you receive your award. And at this point, you basically talk to both your program officer as well as your grants official. So that's really who you interact in the NIH circles when you're thinking about getting a grant funded for yourself. So I use the word program officer multiple times. So what do program officers do? So as program officers, we oversee a portfolio of funded research grants. We advise researchers who are interested in applying for NIH funding. So we have a huge portfolio. We talk to a lot of people. So we do have access to where the institute's priorities are, where the field is heading. So it's really helpful to have a conversation with your program officer. We also do what we call as program development. So we convene, we attend meetings, and we try to address or identify what research gaps exist. And in order to close those research gaps, we actually issue the funding opportunity announcements that I was talking about. We also do what is called a strategic planning. So we propose to the institute that, hey, leadership, look at this. This is an area that's a gap area that needs more funding, that needs more attention. So then the leadership and us have discussions, and that's how funding opportunity announcements get published. So any one of you considering applying for grant funding, I highly recommend finding a program officer and speaking to them. You can discuss with them the relevance of your research concept. You can discuss with them what you think the research activity code should be, and also which funding opportunity is appropriate for your application. So one of the things that everybody in research aims is soaring towards independent funding. And if that is your aim, a program officer can definitely help you accomplish that. There's various ways you can get funding. One of the ways you can start off is by getting on a supplement to an existing grant that maybe your mentor got. Or you can be part of an academic institution with an NIH-supported training program, which is a T32 type of grant. Or you can directly apply for an NIH individual award. And it can be a fellowship award, it can be a K award, or it can be an R research award. So those are the various mechanisms. A lot of people ask me which is a good mechanism. I mean, this is the slide really that answers that. There's really funding to empower you at every career stage, whether you're a pre-doc or a postdoc, or you're trying for something that's a high risk, high reward director's award, or you want to open a cooperative agreement that with the institute, you want to enhance your research by an R15, so on and so forth. It's really the limit is the sky is the limit. But I think instead of focusing on the mechanism, I always suggest my grantees to focus on the idea. What is the idea and what is the question you're trying to address? And once you know the question that you're trying to address, I think that's where you start to build up, and then you find out how much the budget is, and then you find out which mechanism it fits. So it shouldn't really be which mechanism and then find the idea, but it should be the other way around, essentially. So I'm going to skip some of these slides, but I did want to highlight Fogarty International Center because of the audience. As multiple of you are not U.S. citizens, the opportunities available for funding are limited. But nevertheless, that's how people get started, and that's how you build up your research portfolio. So I just took this screenshot out of the Fogarty International Center's open announcements. So if I were a researcher trying to find funding, I would go into these Fogarty programs, click on them, and see what applies to the kind of research that you're doing, and align your application with the funding opportunity announcement that's available. So I did want to, it would be remiss if I didn't discuss how NICHD and India interact. This is just a snapshot of some of the portfolio of funding that we have provided in the last year, or actually the beginning of this year, in 2024. These are some of the applications, and these are some of the investigators who have applied for these mechanisms. Some of them are K, and some of them are an R01. Some of them are a UG3 mechanism. But more importantly, instead of looking at a list, I like looking at images. When we look at the funds that NICHD has provided for research development in India, it has been either to focus in long-term care, where it was towards iron deficiency in pediatrics, or looking at pregnancy and stress in pregnancy, or severe anemia in pregnancy. Some of the rural India was funded for implementation science, contraceptive development, low birth weight areas. So there's really a way we as program officers can look at what is being looked at and how research is happening. And this again helps us to identify what the gap areas are. So for example, there's a health system application, and we can go in there and see what was the specific application that was funded and what else needs to be done. So as proposed, as said earlier, my area of interest is early immune system development and ontogeny. I look at how pediatric immune system works and how it responds to diet or vaccination or environment, and how it actually is programmed to become a robust adult immune system. As I promised, some of the tools and resources. So if you're again considering funding, NIH Report is an amazing open source. You can go in there, you can search for what research area is happening. You can find program officers in your specific research area. There's a bunch of tools that'll help you find which institutes have received awards on that area that you're looking for. Say, for example, you look for pregnancy and malaria. You can find all sorts of applications that were funded. You can find which institutes were awarded, how much money was awarded, what kind of mechanism was awarded, etc. And I'm happy to share this slide deck. There's a bunch of grant writing resources, some tips to help you write a better, stronger grant. Another resource is the NIH Guide. This is where all the latest grants and funding opportunity announcements are published every week. If you are not subscribed, I would subscribe. It'll send you a list of new opportunities that are presented. I am going to go to the end here. So I'm going to stop here with a quote from Eunice Kennedy Shriver. And I really like what she says here. She says that if you don't have an idea that actually materializes and changes a person's life, then what do you have? You have talk, you have Zoom calls, you have telephone calls, you have meetings, but you really don't have a change in the community. And the way you can bring about change in community is by actually doing what you're thinking about. So this is Eunice, and these are the special kids that she always worked for. Thank you so much for giving me this opportunity, and I'll be happy to take any questions in the panel discussion.
Thank you so much, ma'am, for a nice presentation. And your presentation will help everyone at early career stage. Thank you once again, ma'am. Now, the last speaker of this session is Dr. Arku Banerjee, Assistant Professor, Cold Spring Harbor Laboratory, United States. Dr. Banerjee is interested in how the brain transforms sensory inputs into action during natural behavior, focusing on vocal communication in an exotic rodent species from Central American rainforest known as the singing mouse. His research aimed to understand the neural dynamics and circuitry that enables a motor behavior to be used flexibly for social communication. Dr. Banerjee's contribution to the field, including receiving a $3 lakh award to study brain circuit involved in vocalization, which could help explain how the brain drives speech. His work has been recognized through the Clinton-Simon Neuroscience Fellowship. Dr. Banerjee present at the international conference will undoubtedly enrich discussion and inspire collaboration among researchers from around the world. He was selected as ZRF at the Simon Foundation Society of Fellow, New York in 2017, received National Science Foundation travel grant from Greece in 2016, and so many. I welcome you once again, sir. Please continue.
Thank you very much. Can you hear me?
Yes, sir. Yes, sir.
Great. Okay. So thank you for the kind invitation. Thank you for the introduction. I'm really excited to share with all of you our work on the neuroscience of vocal communication. This is Cold Spring Harbor in autumn. This is not how it looks in winter right now. And my lab is somewhere hidden behind these trees. Okay, so I want to start by reminding you that throughout the animal kingdom, many species use sounds to generate behavior. For example, croaking frogs to singing birds, even insects use acoustics to find appropriate mates. We know dolphins make whistles for social interactions. Usain Bolt would basically wait for the sound of the gun to start his 100-meter dash. To us humans engaged in conversation during social interactions. Now, all of this sounds easy, but often it breaks down during communicative disorders such as autism or if you have a stroke. And the neural mechanisms by which animals vocalize and communicate using sounds is not well understood. Now, typically in the lab, the way it's studied in neuroscience is one takes an animal and trains them in a task. So for example, you can have an auditory motor task where there is an animal and then you can play tones of low frequency or high frequency. So it can be like a ting or a tee, and you can train an animal to either lick left or lick right, and the animal learns this. And this is how neuroscientists can go and figure out the responses in the brains that allow animals to do such auditory motor tasks. However, there are limitations. This is a setup where we as experimenters impose our constraints on the animal. What I thought we would do is to follow something known as the Croc's principle, which is that for a large number of problems, turns out that there are some animals of choice on which it can be most conveniently studied. For example, in neuroscience, there's been an enormous diversity of model organisms that have been used for fundamental discoveries. For example, that the neurons make action potential was discovered in squids, or that there is synaptic transmission was discovered in frogs. Currently, there is a diversity lack in neuroscience, and we wanted to correct that. So instead of trying to teach an animal a task to study auditory motor transformations, what we thought we would do is to look at a natural behavior that a particular rodent species does, given its evolution. So we study these Costa Rican singing mice, and these are rodents that have elaborate vocal behaviors. And I want to start by showing you a video of these singing mice. It has sound, so hopefully you'll be able to hear it. Though what you will see here is that there are two singing mice, one on the left and one on the right. The mouse on the left will sing a song, and the mouse on the right will listen to it, turn towards it, and then vocalize in response. Here we go. Great. Did you guys hear it? Probably not. I'm not sure. Okay. Okay. So these animals, they have vocal behaviors that also depend upon how far the animals are with respect to each other. So when these animals get closer to each other, they have sounds that are of different kinds. So if you can't hear it, this is how you can read this. So what I'm going to play is a spectrogram. So you'll have frequency on the y-axis and time on the x-axis, and individual shapes are the sounds that these animals make. Okay, as you can see, they're making a lot of sounds. And now the female starts making a song. The males continue chattering. And they will do this 10,000 times a given hour. So it's a very robust vocal behavior that is modulated heavily by social context. And this is a really remarkable behavior for a rodent. Usually it's unusual. Okay, so on and so forth. So using behaviors of this kind, we want to understand how neural circuits in the brain give rise to such phenomena. If you analyze these vocalizations, what we find is that there generally falls into two categories. One that's called the ultrasonic vocalizations in magenta, and the songs that I have shown you previously in cyan. And they mainly differ in two respects. One is that the ultrasonic vocalizations are short, they don't have temporal structure resembling a song, and they're very soft. And songs have a temporal structure in which it evolves predictably over 10 seconds or so, and it's really loud. And the reason these two modes exist is because they use these two modes at different distances. So for at long distances, they use songs to communicate in the cloud forest of Costa Rica. And then when they get closer, they switch to making ultrasonic vocalizations. In some sense, it resembles the way we flexibly use vocalizations, right? To shout for a friend far away, and then we tend to whisper to our loved ones when we are close. So if you think about the behavior of these singing mice from Costa Rica, it seems to be slightly different from what other rodents do. So on the right here is what a typical lab mouse would do. So you see this ultrasonic vocalization component that is shared, but songs are a behavioral novelty. The way you can think about homologous genes, you can think about homologous behaviors, things that are common across species. And then songs actually represent something that's not there in most rodent species, and it's really a behavioral novelty in the singing mouse. That gives you an opportunity to try to understand how neural circuits produces songs and how that can be different from other rodents, because it gives you an advantage trying to understand how new behaviors might even evolve in the first place. So we start by finding a particular brain area, because the brain is a big place and you need to start somewhere. And we start in the motor cortex. And we identified this motor cortex a few years ago using electrical stimulation. So we'd go and electrically stimulate different parts of the brain and then measure EMG activity from muscles. And there we found a hotspot of neural activity. And we knew that this brain region might control song. The way we tested it is I pretended to be mouse one and started playing back songs over speaker. And then on the left, you see that the animal basically gets fooled reasonably and starts responding to playback. And among 20 such playback songs, maybe it responded to 18 of those. So a high degree of response. But if you inactivate this motor cortex using a drug, that behavior falls apart. So we know this actually causally responsible for this vocal turn-taking behavior. Then we wanted to get to a more interesting question, I think, which is that, okay, so if the motor cortex is involved in singing, and I told you song is a novel behavior that does not exist in the lab mouse, are there differences in the way motor cortical neurons connect and wire up with the rest of the brain? So what we are testing is a hypothesis that perhaps there is structural modifications of motor cortical neurons in the singing mouse that allows them to express this novel behavior. And it's a hard problem because if you just look at brain slices, they look remarkably similar. So it's not like, a new brain region has come up or the brain looks very different. It has to be in some details of how the connectivity is set up. So we had a few hypotheses. Perhaps there is a novel projection, right? So maybe motor cortex projects to a different part of the brain that does not exist in the lab mouse. Or perhaps there aren't any such novel projections, but the connection strength is higher, right? So, and these are the models that we want to distinguish. So the way we do this is we inject viruses in the motor cortex. The neurons express GFP, sends the GFP along the axons, and we can do 2-photon tomography through the entire brain, trying to figure out where the axons are going. So these fluorescence signals are where all the axons are going from motor cortex through the rest of the brain. And we can do this in lab mouse and a singing mouse, and we color coded them. And the fact that you can't see two colors basically means that we didn't find any qualitative differences in brain-wide projection targets. It's not as if there is a novel target or a new area that has suddenly evolved. So perhaps there is a change of connectivity strength or connectivity statistics. And the way we attempted to find this out is by doing neural not anatomy, but by doing sequencing instead of tracing. So the way this works is we inject a viral library of oligonucleotides. So each bar is basically each oligonucleotide is about 20 nucleotides long. So it can express a unique barcode. There can be 4 to the power 20 unique barcodes in a viral library. So you inject virus, each neuron takes up a particular barcode, and then that barcodes get sent throughout the axons. So then all you need to do is to cut up different portions of the brain and just do a sequencing, right? If you sequence, then you know whether a particular barcode and hence a particular neuron was present in these target areas. So with this, where, typically with light microscopy, you can maybe reconstruct about 10 neurons connectivity in a given brain. Here we can do about 5,000 neurons at the same time. So here what I'm showing you is basically thousands of neurons as rows, and their connectivity as color coded for different brain regions, which are in columns. And we can do this for the lab mouse. We can do it for the singing mouse. We recover the generic set of neurons that we expect, things that go within the cortex called IT neurons, things go to the brainstem called the PT neurons. So overall, again, it looks very similar. But here we can now quantify because we have the exact amount of barcode per neuron. And if we do that, what we find remarkably that out of all the brain regions that we tried, there's 2 brain regions where there seems to be a factor of 4 increase in the connectivity strength in the singing mouse. And those 2 regions are auditory cortex and the midbrain region known as periaqueductal gray that through last 40 years, people know have been involved in vocalization. So suddenly we find that there are these 2 areas where motor cortex connects to that have about a factor of 4 increased strength of connections in the singing mouse. So what we think is going on is that the way this behavior has evolved is not by generating a novel projection, but by increasing the projection strength of specific circuits in the brains of the singing mouse compared to its ancestral rodent species. Okay, this is about structure. What about function? Maybe there are functional differences in how motor cortical neurons respond to either songs or calls. So for this, we have to do an experiment that's slightly complicated. So what we need to do is we need to insert electrodes in specific brain areas of the singing mouse, design it such that it can be held on top of the brain along with the hardware, and the animals have to be able to carry this and still make these vocalizations. And when we do that, we find that there are many, many neurons that are responsive during songs. So some neurons that are going up before the song, some that fire at the end of the song, and then some neurons that are persistently active throughout the song. And we can check and figure out how exactly it varies with songs. So as the songs become longer and longer, the neural activity actually stretches like a rubber band, as you can see in these example neurons. So neural activity stretches as if, that's how they control how long the song is going to be. So we have discovered an algorithm by which the song durations is controlled by activity in the motor cortex. And the last thing I want to show is basically what we think is going on is that motor cortical neural activity hierarchically controls vocalization, much like it does in humans as well, that it controls subtle things like how long the song is going to be, when does it need to start, when does it need to stop. It does not control the basic act of singing, but modulates it on top of it. And more importantly, these neural activity was specific for the songs and not for the ultrasonic vocalizations, which are the ancestral behavior. So it means that neural activity in motor cortex is actually specialized for the behavioral novelty. So what we think is going on, and this is the take-home message, that we have discovered behavior in a novel rodent species where we can confidently say that this rodent has a new behavior, a novelty, and we have localized a circuit for that novelty in the brain. And we think that they're both structural as well as functional specializations of motor cortex that enable this animal to generate behavior of this kind. So in general, I think it gives us an overview about how neural activity controls certain natural behaviors and how new behaviors might even evolve from ancestral circuits where you don't express that behavior. I would like to thank the people in my lab, Cliff, Mike, Emily, and Yaman, who did the work, my collaborators, funding source, the core facilities at CSHL, and thank you for your attention.
That was a great talk, Arko.
Thank you.
Really fun to listen and very informative. Thank you.
This was the first time I have given a talk after midnight, so I was worried
But
for a moment.
you sounded as passionate, I am sure, as you sound, during the daytime.
Espresso will do the trick for you.
Yeah.
Hello, Arko.
Thank you.
Thank you
Hi.
so much, sir, for
You're
such
welcome.
a creative presentation. Thank you once again, sir. And now it is my responsibility to thank every speaker on behalf of Amity Institute of Biotechnology, Amity University, Madhya Pradesh. And today I have found that every speaker finished their talk within time. That's very great, sir. Thank you so much. Now the session is open for the panel discussion, and a student can text in question and answer box. Everyone, please open your camera, and the session is open.
Is that okay? I actually have a question for Dr. Banerjee.
Please.
This is so fascinating because we have some portfolio that includes nonverbal autistic children. And some of the research collaborates with the fact that memory and sounds are related. So I'm wondering if your lab mice remember less than your singing mice, and if the sounds actually have some sort of cognitive effect on the singing mice.
It's a great question. So I think, you have to think about whenever animals do a particular behavior, if you think about why is it that this particular animal has evolved this behavior in the first place. So these animals are from Costa Rican cloud forests, which basically, if you go there, you'll find that there's a lot of vegetation. So they can't see long distance. So they've developed a way of communicating long distance using audio. Now, why that might be useful, you might be actually, it would be very beneficial if they can identify individuals using their, appropriate sounds. So I think that gets to what you were asking about. I think we have evidence that these animals use sounds to identify individuals, which obviously means that they have to be able to store the sounds in their memory and access it online in order to sort of figure out how they should respond. If it's a foe versus a friend, the way they would respond is very different. And I think your original point about like ontogeny, the development of this behavior is very interesting. So you have a divergence. When does it happen as a function of development? So we plan to look through the entire process of ontogeny and see where are the divergences. At what point would the circuitry diverge and then this new behavior starts expressing? Great question.
I have another question for you, Arko. So is this so this looks like is an evolutionary behavior, but is it also a learned behavior among mice? Can lab mice learn to sing along with the singing mice, or the singing mice lose their singing if they are bred among the lab mice?
Great question. So I think, it's quite natural to think about the issue of learning because, for example, a bird, a zebra finch, which many people study, they actually learn their vocalization from their fathers. We tend to learn vocalizations from, people we interact in our childhood. Turns out that in this particular species, in all rodents so far, there is no evidence for vocal learning. Okay, so we don't think that this particular vocalizations is actually learned. However, there's a large degree of social modifiability. So don't need to learn in order to know how to appropriately respond. So they can change their vocalizations on the fly in a fast and flexible manner. And what we don't quite know is whether this ability to flexibly communicate requires auditory experience growing up. So that's an experiment that's part of the ontogeny question. So if you deprive an animal, say, for example, auditory input during the process of development, we expect it to still sing a song, but not to be able to vary the various features of the song that they do during flexible communication. But sort of that's the general nature of the hypothesis that we want to test.
Thank you.
Okay, thank you everyone. Any question from anyone's side? Okay, now it's time of group photograph with all the respected speakers. Please everyone open your camera.
You may have to call Dr. Naresh.
Yes, sir.
Yes,
Mishra sir is calling.
I'm trying to call.
I was actually thinking about, giving a talk about vocal communication where it's one-sided.
Yeah.
Right? Like it just tells you the importance of being able to do bidirectional communication.
The Zoom jokes were aplenty in your lab. Can you
Huh?
hear me? Can you hear me?
Yeah.
The Zoom jokes.
Yeah.
He is not able to hear us, ma'am.
Yeah.
Okay, for a group photograph, please, everyone. Okay, thank you once again, everyone. Okay, now next session will continue by Dr. Asha. Please, ma'am.
All right, I'll take leave. Thank you very much.
Thanks all once again.
Thank you all. Thank you.
Thank you very much, everyone.
Great panel, guys. It was really fun to talk to all of you. Thank you.
Hello, everyone. So finally, we are moving forward to the session 5, genetic engineering and bioinformatics. The chairperson of the session 6, Dr. Muthu, and the co-chairperson, Professor Dr. Vinay Dwivedi, and the invited talk delivered by the Dr. Shushma Chauhan, Dr. Pankaj Rai. So first of all, I would like to call Dr. Shushma Chauhan. Dr. Shushma Chauhan, visiting scientist, Dongkuk
Dr.
University,
Asa, she is
South
coming.
Korea.
Dr. Asa,
Yes, sir.
just 2 minutes. She's joining.
Ma'am. Okay.
Hello, ma'am. Good afternoon.
Good
Ma'am,
afternoon.
could you give me some time? Actually, just I came from meeting. I have to. I mean,
Okay, no
join
issue,
with
ma'am.
another.
No issue. No issue.
Yeah.
I'm just introduce you right now. Huh?
Yes, ma'am. Yeah. Thank you so much.
Nice. Dr. Chauhan, shall we start?
Hello, ma'am. Can you hear me? Dr. Susma here.
Hello, Dr. Sushma. How are you? This is Dr. Vinay Dwivedi, this side.
Yes, sir. Good afternoon. I'm good, sir. How are you, sir?
I'm fine. I'm fine. So now you are ready?
Sir, give me just 2 minutes, sir. I'm sorry to say because actually I had all sudden meeting with professor here, so I just came down.
Yeah, it's okay. No
Now
problem. No problem. So just
yeah.
set your things and then let me know so that we can start.
Yes, sir. Just give me 5 minutes, sir. Within 5 minutes,
Sure.
I'm connecting with the computer actually.
Right.
Yes,
Right.
ma'am,
No
you
problem.
can.
No problem.
Thank you, sir.
So, first of all, I'll welcome to Dr. Sushma Chauhan. She's a visiting scientist, Dongkook University, South Korea, and assistant professor and research coordinator, MIT Institute of Biotechnology, MIT University, Chhattisgarh. Dr. Chauhan academic journey is marked by the exemplary achievement and culminating in PhD in biochemical engineering from Dongkook University, South Korea. Prior to ma'am doctoral studies, she obtained the master degree in biotechnology
Zoom में.
from Guru Ghasidas University, India, and the bachelor degree in the microbiology from the same institution. Dr. Chauhan dedications to education extend beyond the classroom as she has taught a diverse range of courses and provided valuable services to the students and the host institute. Her expertise in areas such as advanced plant biotechnology, vaccine technology, genomics, proteomics, and computational biology has enriched the academic experience of her students and contributed to the advancement of the biotechnology field. We welcome you, ma'am. So you can please deliver your lecture by sharing the screen.
Thank you so much, ma'am. I'm still connecting with my office computer actually.
Okay, ma'am,
Just
you'll take time.
give me a second. Yeah,
Sure,
thank
ma'am.
you. Actually, all sudden professors.
Ma'am, please unmute yourself, then we will able to hear you. No, ma'am, you are not audible, ma'am. Still, you are muted.
Yes, ma'am, I'm sharing the screen. Now only
Okay.
I connected with another system.
Okay.
So,
Yeah, sure.
okay, hope you are able to see my screen now.
Yes, ma'am, it's visible.
Thank you, ma'am. So first of all, I would like to thank MIT University, Gwalior and Professor Raghavan.
Ma'am, we are not able to hear you. Hello.
Hello.
Yes, ma'am. Right now you are audible.
Thank you so much, ma'am. Actually, today I'm using my colleague office actually because my lab is little crowded, so I thought of using this so that the noise will be not there. So little new for me. And thank you. With that, I would like to thank the entire team and Professor Raghavan sir, who actually invited me for the talk. And it is my privilege to deliver the talk in this conference. So with that, I would like to start my talk. And in my talk, basically, I'm going to talk about the classical protein with the modern approach, and that is why I have chosen the title, that is the rings of innovation and the circular protein in the modern or contemporary biotechnology. So my talk will be going to cover the basically circular proteins. And as a biotechnologist, we are known that the structure of protein basically, for especially for the students and the audience who are basically working in the biotechnology, they will know that the protein are having two termines, that is the N terminal and the C terminal. And these proteins are called in a simple term that linear protein. But what exactly a cyclic protein or circular protein is? So basically, circular proteins is a unique proteins which is a class of biomolecules that characterized by their closed loop structure architectures and have emerged very pivotal players in modern biotechnology. So the question is, why protein? Why this circular protein? So because of their circular architectures, it imbues them with the exceptional stability and resistant to toward the higher stability against the proteolytic degradation, high thermal stability, and acid and alkali resistance. Also, it possesses the higher chemical stability. Moreover, it enhances the turnover activity. And also, because of this tremendous versatility, these actually making them ideal candidates for the various biotechnological applications. And therefore, the stable peptides can actually, or proteins will be broaden their uses in various fields, like designing the protein-based or peptide-based stable drugs. So the applications, now we have to talk about the applications, because ultimately we are going to explore the this special proteins. So the applications of the cyclic proteins are in pharmaceutical industries, which improve the say pharmacokinetics and pharmacodynamics of the drugs, like insulins, antimicrobial peptides, growth hormones, peptides, and protein-based vaccines. So right now I'm working in a development of antimicrobial peptide with the in vitro systems. So we are employing these cyclic proteins. So another applications is the industrial applications. Like, we are talking about enzymes. These are the list of enzymes which we are regularly working, like amylase, lactase, pectinase. We are talking about how to isolate the enzyme, how to employ those enzymes into the applications. But what about the stability? What about the stable conditions? We have to think about beyond that things also. So these cyclic strategy can also add on in the stability of those industrial enzymes. The next applications is in the biophysical properties and specifically in the environment. So these stable proteins like for example, PATase or horse radish peroxidase, those are the peroxidase-based enzymes. Those are well known for the bioremediations and for the wastewater treatments and also for the sequestration of the heavy metals. So those stable enzymes, with the help of nanotechnology or nanobiotechnology, we can make a very stable catalysis and we can use for the environmental cleanup. Another example or another application is in the diagnostics. So diagnostics, again, we have to think about the stability right from the drug or right from the use of. So the best example here I would like to give is the horse radish peroxidase or glucose-based enzymes, which we are using as a biosensor or even for the bioconjugation of those enzymes for the Western blotting for the antibodies, because we are regularly, like in my lab, we are regularly using those antibodies, and all the secondary antibodies are bioconjugated with the peroxidase based or alkaline based. So those stability also we have to think about where we have to store, like refrigeration or -20. So if those things can be cyclized and we can use bioconjugation, then we don't have to really worried about the storage condition and all. So the example is also in a COVID testing kits where we are using enzyme-based biosensor only. So we know the storage condition and all. We have to again think about those storage conditions. So these cyclic peptides and proteins are having tremendous applications. Hence, naturally, this system has been adopted just because of their superiority of the cyclic proteins. All the natures already from the higher molecules to the lower molecules, because of the configuration to protect the host cells from the diverse like stress or from the microorganisms. This system has been already adopted, and we know the antimicrobial peptides is a key components of innate defense molecules against the bacteria, fungi, and viruses. Hence, these cyclic antimicrobials were highly resistant toward the pathogens or microorganisms proteases. So the question is how to synthesize the cyclic proteins or the peptides. Well, there are chemical methods which are available, and that chemical methods are really not economically feasible, and also it is having limitations, like we cannot go for the more than 18 to 20 amino acid residues for the synthesis, and it always leave with the one auxiliary sequence that sometimes hinder the sequence, or sometimes it give the steric hindrance to the activity in the further downstream part. Another method is the biological methods. Moreover, in the chemical method, we have to think about long time, storage of the chemical in the chemical, because the chemical can over a time do the reaction and enzyme activity can be lost. So the another method of this cyclization synthesis is the biological methods of the cyclization, which include the enzyme-based that is known as a sortase-mediated cyclizations, butylase-mediated cyclization, and genetic code reprogramming for the unnatural amino acid incorporation and synthesis of the cyclic proteins, and of course, intine-mediated cyclization. So here in our research, we have focused in the intine-mediated cyclization, and it is having more advantages over the chemical method and other biological methods, because these are the genetic elements which are naturally occurring, and it is having autocatalysis. So we don't have to treat with any specific chemical or anything. It has the auto splicing phenomena. So the advantage over the method is no need to worry about the size limitation, as well as it is having high scale production and cost effective, and also we can do the in vitro as well as in vivo. So these are the now what is intine molecules. So I would give a brief introduction that what these intine molecules are. Well, these intine molecules are genetic elements which are embedded into the bacteria genomes, and this has been already found in the several classes of bacteria and in the A, you can see this is the structure of intine, and the green beta sheet and helicals, you can see that is the C intine, and this is the N intine, and these intines are the very important molecule of DNA polymerase 3. So we all know what is DNA polymerase 3, but the activation behind the DNA polymerase is actually the intine molecules. When the DNA polymerase go into the heating, heating, then only this intine molecule is spliced out and it remove the blockage, and then only other factor can go and it can activate the DNA polymerase. So these intine molecules are having it's well known for the just a piece of genetic elements which don't have any functions. But now the research is coming out to engineer those intine molecule and explore as a biotechnological tool to make the protein synthesized in a form of cyclic, or to study the protein-protein interaction or peptide-to-peptide interactions. So this structure B is actually the multiple sequence alignments of these intine from different species, and those green arrows are showing that from where we can do the splitting, and also for the protein-protein interaction analysis, or changing the amino acid into the different amino acids. So now green fluorescent protein, a very important fluorescent protein for the any biotechnologist or any scientist who are working in the protein engineering. So similarly, we also opted the green fluorescence protein, and we know the green fluorescence protein is like a highly barrel seed beta with the beta seeds, right? So as all proteins or all enzymes, this green fluorescent proteins is also having N and C terminal, that is like basic structure of the any linear proteins. So how cyclic protein looks like? So this is the cyclic proteins after the connection of N and C terminal via the peptide bond or any chemical method or enzymatic cyclic method. The structure will be look like that, which will provide the structure stability as well as the rigidity to the any proteins. Now the cyclic proteins, how it look like and how we are going to detect it. So there is a high throughput high throughput proteomic tool which are known as a LCMS and MALDI-TOF. This protein can be detected. Now for the for this method, we need the cyclic protein in a pure form. Very challenging task. Number two, this method itself is a expensive method. So we have to also look for the solution where we should not, low cost with high productivity. Always we talk about that. That's what we want to do in the research. So the question which when we started this protein engineering things, we thought that how we are going to detect in a very feasible way in a in-house detection system. So we when we do the cyclization, genetic engineering, always there is a 50% possibility that half of the protein will go into the hydrolysis part, half of the protein will go into the cyclic parts. So overall in the entire system, entire solution mixture, you will have both the mixture. Now we have to think about the techniques to identify at the same time how we are going to purify the counter cyclic protein from the linear counterpart. So we had a big question mark because we cannot go each and every time for the MALDI-TOF as it is a very expensive method. So we came with another idea that is the epitope engineering. So epitope engineering is actually we did with the CMYC tag. CMYC tag is a tiny 11 amino acid tag where we just did the engineering by splitting. And after splitting, we know that the antibody, the simple ELISA method, that if there is a complete tag, any antigenic tag is there, then only the antibody is going to interact. Other than that, if there is any mutation or any splitting is there, this antibody is not going to interact with the antigens. So antigenicity of antibody and antigens will be lost. So similarly, we adopted that idea and we choose MYC tag for the genetic engineering. And then there was some specific requirement for using the intent-mediated cyclization, that how we can use so that it will give the perfect, cyclic product to us. So the first thing, it should have the ability to use both in and serine, cysteine, and threonine is well known for the good nucleophilic group and good living group. So these things should be there. So luckily in our this tag, we had went through the different mutation and selection of the best tag. So we this is look like um, uh, wild type MIG tag, and this is the linear GFP. How, if the cyclic GFP will be there, how it is going to detect through the anti-MIG antibody. So if the product in the mixture is a linear mixture, then obviously it is not going to bind with the antibody, and we will not be able to see any detection into the Western system. But if the product is having circular and the both in has been connected, then only the antibody will be able to detect. So this article we published in 2018 and 2013, two different paper. And the strategy of engineering this split tag was very simple, but of course we had done lot many genetic engineering and screening. So the splitting would be like the target protein. Here we choose as a GFP as a target protein, and we found the first amino acid, as I mentioned in my previous slide, it should be serine, cysteine, or threonine. So the X could be the serine, cysteine, or threonine, and C would be start with the cysteine. So this is the cleavage point. And then we know that the this formyl methionine can be when protein translation happen, formally that formyl methionine is the first methionine of the any protein which start from the decoding. So this formyl methionine can be removed out in the system during the in vivo expression due to the map that aminopeptidase or peptidyl drop off. So that we will in that way, how this X of this any 3 amino acid will be exposed out, the nucleophilic will attack, it will go for the NS acyl shift, and then it will subsequently go for the nucleophilic attack from this X position to the another cysteine position, thiol position. And then it will be like it would have two chances. Number one, hydrolysis. If the product is going into the hydrolysis and if water molecules is occupying before the peptide bond is synthesized, then it will leave for the linear product. But if the perfect nucleophilic attack has been taken care and the water molecules occupancy is limited there, then it will go for the backbone cyclization of the target with the full tag. So we engineered that tag, and then we also demonstrated the cyclization as well as different genetic mutation over the tag, and we found E-MIG. E is actually, we engineered the MIG tag, wild type MIG tag at the at position from originally from glutamic to the cysteine. So we have given the different names. So in our research, we found this at E at MIG C was the best results. And then we synthesize in-house bacterially expressed antibody. Again, that was a different project where we was wanted to bypass the commercial available resin for the purification for the industrial use, and we transfer this technology to the Korea CJ Chemical, and there is one antibody production company. So we designed this, we immobilize this antibody into the activated thiol bead, and how this thiol bead can be picked up only the cyclic GFP, not the linear GFP. So in the mixture, how the purification will be takes place, which we published in 2013 in Analytical Biochemistry. So we proved this one, and then also we started our journey for different peptides. Like as I said, we are working in antimicrobial peptides also, and other complicated industrial and environmental important enzymes. So we proved this concept, and we also the proof of concepts was proved in terms of stability assay, and we proved that the backbone cyclized protein would be more stable than the linear counterpart. as you can see in the picture A and B. So now thing is that improving the stability of pharmaceutical, that previous slide was the demonstration of how we are started our journey, and then here we are going to talk about the any pharmaceuticals or industrial enzymes, how we can from the model protein to switch to the classical proteins. So improving the stability of pharmaceutically important proteins via end-to-end cyclization. So we has chosen the horseradish peroxidase. So what is horseradish peroxidase? Basically, horseradish peroxidase is a heme-containing enzyme that catalyzes the oxidation of wide range of aromatic compounds, and it has been isolated from the a plant that the plant is known as a Armoraceae rusticana, and it is having different applications, like wide range of applications, including medical diagnosis, biosensing, in bioremediation, medical diagnosis, more importantly, immunodetection, because we are regularly using bioremediation and immunodetection. And this HRP is conjugated with secondary and antibody. So how we are getting enhanced specific signals. So these are the very this enzyme is a very applicable, especially in the analytical laboratories. So the thing was a soluble expression, because this enzyme, as I said, is from plant source. So as a previous result reported that this is a highly soluble, insoluble protein, and that our challenges started with working with right from the soluble expression, because for the cyclization we need the cyclic protein, not the insoluble proteins. So soluble expression and purification of recombinant HRP. So we made a construct with the T7 strong. The T7 promoter is known as a very strong promoter, and we choose the PET vector as a vector system, and we make this construct. When we started our work, we end up with the totally all the protein was going into the insoluble fraction, which is no use for us. Then we started looking for the possibility that how we can engineer again this enzyme with any other partners, so that we can make this protein into the soluble fraction. So we choose the phosphoglycerokinase. The reason behind choosing the phosphoglycerokinase is that this PGK is a very important enzyme in the glycolysis pathway, and it is found in E. coli, and highly soluble and highly stable protein. So it will our hypothesis was that if we will use this PGK from the E. coli, it will also during the expression, it will allow the down gene to express in a soluble form along with the PGK. So we make a construct like that, and then when we engineer this PGK with the HRP using GES is a glycine serine linker to make flexible loop between those two big proteins, then we found that the strategy was work, and we could able to get a very highly soluble HRP. And then this HRP, we went for the in-house activation, as we know that peroxidase is a complex enzyme which need many factors for the activation, like calcium chloride and hamine and disulfide bond, very important. So those things we did, and we found that our HRP is very active enough to go for the further step of the cyclization. Along with that, we also engineered the E. coli, because we wanted to bypass adding of extra heme into the activation process, but we wanted to activate the ferrochylate genes like a co-expression in E. coli. So we engineered we blocked the transporter of hemine into the system, and we make the a co-expression construct where we wanted to express the ferrochelated genes, which is a gene, very important genes, which use ALA. ALA is amino linoleic acid as a precursor molecules, very important molecule for heme biosynthesis. So system itself can make a heme, and that heme can be incorporated into during the expression of the HRP itself. So it is kind of in vivo hemine incorporation into the expressed HRP in the system during the protein translation. However, when we did this experiment, we successfully demonstrated the co-expression and a good expression of PGK-HRP, and also when we did the activity assay without hemine also, we had a activation, but the yield was very low. So because of yield, we could not say that it is 100% good hypothesis, but anyway, we tested that. Incorporation was there, but other engineering right now I am doing, so the work is going on. So then because we had a well enough soluble HRP, so we shifted to explore the split intine system to get the cyclic product. So then we started making another expression construct using split intine. So as I said, this intine is a whole intine molecule. When we split out the intine into N and C part, it can be joined together later during the autocatalysis or splicing mechanism. So the same cassette we use, and we just engineered the half of the N intine part into the N terminal of the target, and C terminal of the target was having C intine. And when we did the expression, we confirm into the Western blot that the product is giving 50% of the cyclic and 50% of the linear product also. So later on, we did the second primary and secondary purification. So as you can see in our construct, we are having two different tag. One is a emic tag and another one is a that 6x his tag. His tag we kept here for the primary purification using affinity nickel affinity chromatography, and emic we as detection as well as for the purification, as I earlier mentioned, that is that we did the immuno purification specifically. So from primary purification product, we did the secondary purification product using the immuno anti-mic anti-resin. So we successfully able to purify the cyclic HRP out of the linear mixture, and activity was also half 50% as a commercial HRP, plant derived HRP. So then later we further conducted the experiments to study the stability of cyclic HRP versus linear plant-derived HRP. So we tested different conditions, for example, chemical denaturants, and we know that guanidinium chloride is a very harsh chemical in the for the protein engineer specifically. Urea and guanidinium chloride completely denature the structure of protein. It completely kill the structure of protein. When we introduced the HRP and that cyclic and linear into 3 molar, we found that the cyclic HRP was more stable than the linear one, similarly in high concentration like 4 molar, and unfortunately in 5 molar within 5, the cyclic was stable till 5 minutes, and then it lost the activity. And when we went for the heat stability in different heat condition, we found that the cyclic HRP was more stable than the linear counterpart. And pH also, it shows extraordinary activity, because we know that this optimized condition of the activity of the HRP is pH 5, but the cyclic HRP was active even in the low pH, that was the pH 3. And this gives the new finding in our research that even cyclic HRP can be stable enough into the low, low pH also. So this cyclic protein and peptide work we demonstrated with different peptides also, like RGD is one of the peptide, a very important peptide. We synthesized genetically in the lab because we was working in the matrix metalloprotease, and we know that aging problem. So Korea is obsessed with the anti-aging things. So we got one project from the commercial cosmetic company, and we wanted to see the apply these peptides into the anti-aging cream. So we want matrix metalloprotease is a basically very complex enzyme in the collagen. It's a collagen. It's degrade the collagen of the skin, and if your layer of collagen will be degraded out by this matrix metalloproteases, then we got the aging problem. So we started this work keeping this RGD as a blocker. So we designed this RGD and we did the cyclized of this as RGD using the intermediated system, and we successfully found that this RGD cyclic RGD can be act as a blocker for the matrix metalloprotease enzymes. Later on, we transferred this technology to the CJ company for the cosmetic purpose, and we published the data into enzyme and microbial technology. So with that, I would like to conclude my talk, and the conclusion of the talk is the cyclic proteins and peptides can be a next generation exclusive, it will be like elusive biomolecules in the modern biotechnology words. And also the cyclic proteins and peptides enhance the biophysical property by increasing the entropy and decreasing the enthalpy, which subsequently provide the thermal and structural rigidity to the protein. Also, few methods have been developed to produce cyclic proteins and have limitations like cost and also size limitations. For example, chemical that is known as a native chemical ligation. The chemicals are very, very expensive, and those all chemicals should have protonation and deprotonation capability. So that's why the chemical cost is very high, so that we cannot use for all the industrial important proteins. So we have to look for the cost effective strategy. Therefore, we developed an enzyme-based strategy to cyclize any proteins in which can be used for the heterologous expression, as well as we develop the soluble expression of the complex protein like HRP to the recombinantly produced into the soluble fraction. Subsequently, we also demonstrated the split tag engineering, and we expressed that split tag into genetically. We fused into the genetic system, and then also we concluded that it used for the detection as well as for the purification of the cyclic proteins, and how this can be this method was cost effective, and we can produce in a large scales also. So we don't need to worry about the buying from the commercial from the company, which is cost when I started, I bought $2,000 just 100 microliter of the bead for the purification. So we have to that's how we started our journey to demonstrate another experiment, another project for the single chain antibody preparation of CMYC. And then we have successfully produced the cyclic HRP using our strategy, and successfully shown the enhanced biophysical property by demonstrating the stability characterizations using different chemicals. And this strategy can be easily exploited in the modern biotechnology field to improve the biophysical properties of peptides, and also it will have the tremendous application in various biotechnological sectors. And also it will help us to understand the versatility and structural intricacy of circular proteins, which will open new avenues for the development of next generation biotechnological tools and therapeutics as well. So with that, I would like to conclude my talk, and I would like to acknowledge the National Research Fund agency that Korea to support our research fund, and Dongguk University and Protein Molecular Engineering Lab. And I would also like to thank Amity University, Chhattisgarh, who provided me the sabbatical leave so I could successfully continue my research here. So with that, I would like to thank you. And this is my research team here, and my professor, actually he's my PhD supervisor as well. So on behalf of my protein molecular engineering lab, I would like to thank and especially to the entire team of Gwalior campus, and faculty members, all the dignitaries, and Amity University for giving me this opportunity. So thank you very much, sir and ma'am. So I hope I have not taken much of the time.
No,
So
ma'am. Thank you so much, ma'am, for very informative and thought-provoking lecture on the cyclic protein and the applications of protein-based drug. It's
thank you
very
so much, ma'am.
informative, ma'am. It's very nice, ma'am. Ma'am, question answer session we will discuss in the panel discussion time.
Yes,
So
ma'am,
I
sure.
would like to call our next speaker. Ma'am, please unshare your screen. Yeah.
Yes, ma'am. Thank you so
Uh,
much, ma'am.
okay, thank you, ma'am. Next speaker is Dr. Muthu Trivengadam. Sir, are you there? Dr. Muthu, are you there? Sir, Dr. Muthu, are you there? Shortly, we will connect with the sir. So it's a 5 minutes pause only. Okay, so now let's start the next speaker of the last session, Dr. Muthu Theruvenganam. He is an associate professor, Department of Applied Biosciences, College of Life and Environmental Sciences, Konkuk University, Seoul, South Korea. Dr. Muthu obtained his PhD in botany, specialization in plant biotechnology from Bharatidasan University, Tiruchirappalli, Tamil Nadu, India, under the supervision of Professor N. Jayabalan. His doctoral research focused on the in vitro culture and agrobacterium mediated genetic transformation studies on Monodica charita and the Momodica dioica. Dr. Muthu's scholarly contributed are evident from his extensive publication record, over 250 research article, book chapter to his credit. As an esteemed member of the scientific community, Dr. Muthu served on the editorial board of prestigious journal and has organized a special issue on various topics related to the plant biochemistry and nanotechnology. His patented invention further demonstrate his innovative approach to research and technology development. I welcome you, sir. Please connect and share your screen, sir. Sir, are you there? Yes, sir. Sir, your slide is visible, but you are not audible, sir. Sir, your speaker is on, sir. Hello. Dr. Muthu, is there any problem, sir? So we are not able to hear you, sir. Sir, please unmute yourself.
Okay.
Yes, sir. Now you are audible, sir. Please.
Okay, it's fine. Okay, thank you. I'm sorry. Okay, good morning to all. Thank you for giving me this wonderful opportunity. Is it okay, ma'am?
Yes, sir. You are audible, sir.
Audible. Okay. Thank you for giving me the wonderful opportunity to deliver my talk in the international conference in Amity University. I would like to
Just mute your laptop. Just mute your laptop. So
Oh my God.
Just mute.
Is there some problem?
Just mute the sound of the speaker.
Yes.
Or lower the laptop volume.
Is it okay now?
Yes, sir. Now it's okay, sir.
Now it's fine. Okay, I'm sorry.
Yes, now it's fine. No issues.
Okay. I'm sorry. Okay, good morning to all. Thank you for giving me the wonderful opportunity to deliver my talk in this international conference in the Amity University. I would like to express my sincere thanks to the chairperson and all the academic faculties involved in the conference. My special thanks to Professor R.K. Misra, who invited me to this conference. Once again, thank you all the members who are present here. Now I would like to deliver my talk. The topic is exploring the infinite frontiers of nanobiotechnology, innovations, challenges, and applications. Nanotechnology in the science, engineering, and manipulation of materials on the smallest scale, typically involving structures and devices with dimension of 1 to 100 nanometer. Nanobiotechnology is an interdisciplinary field that merges nanotechnology with biotechnology, enabling the manipulation and application of materials at the nanoscale for advancements in medicine, agriculture, and energy. Application of nanotechnology. Nanotechnology using various applications like agriculture and food, cosmetics and paints, biomedical and drug delivery, optical engineering and communication, electronics, metallurgy and materials, defense and security, energy storage. Types of nanoparticles. Metal nanoparticles such as gold, silver, magnesium, titanium, zinc, iron, and platinum. Metal oxide nanoparticles, magnesium oxide, titanium dioxide, zinc oxide, and iron oxide. In our laboratory, we are focusing metal nanoparticles and metal oxide nanoparticles. We are synthesizing and characterization, and we are doing biological activities. Polymeric nanoparticles, polylactic acid or polyethylene glycol. Lipid nanoparticles like liposomes and solid nanoparticles. Carbon nanotubes. These cylindrical structures made of carbon atoms exhibit extraordinary mechanical strength, electrical conductivity, and thermal conductivity. Quantum dots, semiconductor nanoparticles with unique optical and electronic properties such as size tunable, fluorescence, and high photostability. Ceramic nanoparticles, silicon dioxide and alumina exhibit high mechanical strength, thermal stability, and chemical inertness.
Sorry, Muthu sir, to interrupt you. Actually, your slides are not moving. Still, we are in the first slide.
Oh my God.
Yes, sir. Sir, you are mute actually. We are not able to hear you. Sir, you are mute, sir. Please unmute yourself.
So on the mobile phone, please unmute yourself.
Is it okay, ma'am?
Yes, sir, it's okay, sir. But slide is not changing, sir, still.
Yes, I don't know how to.
Sir, you are using the laptop for changing the slide, huh? So please change the slides on laptop. And don't mute yourself on mobile phone, sir. Yes, sir, now it's visible.
Is it okay, ma'am?
Yes, sir, it's okay.
Yeah. Okay, synthesis of nanoparticles. Chemical methods like chemical reduction, sol gel synthesis, and precipitation. Physical methods, laser ablation, sputtering, and evaporation. Biological methods, microbial synthesis, plant-mediated synthesis, and enzymatic synthesis. In our laboratory, we are using biological methods, using various medicinal plants, plant extracts, and microbial synthesis, like bacterial culture we are using for green synthesis of nanoparticles. Because green synthesis of nanoparticle, eco-friendly, biocompatible, cost-effective, controlled synthesis, scalability, reproducibility, versatile, diverse, sustainable, reduced environmental impact, easy accessible, precise, natural resources. So we have synthesized many metal nanoparticle and metal oxide nanoparticles from various medicinal plant extracts. And now this slide showing important medicinal plants, medicinal tree, Abrus precatorius. This is a medicinal tree for using for biological synthesis. This is, we are using the plant bark. This is the powder. This is the liquid crude extract. So magnesium solution, and so reduction of the nanoparticle, magnesium oxide nanoparticle. This is the magnesium nanoparticle powder. This paper recently published in the bioengineering journal. So after this biosynthesized nanoparticle characterization, characterization of magnesium particle nanoparticles, we are characterized XRT, FTIR, SEM, and TEM. XRT for confirmed the particles crystalline nature, and FTIR for wavelength, the presence of biological molecules. SEM and TEM for the size confirmation, and UV also we confirmed. So this slide shows the biological activity for the green synthesis of magnesium oxide nanoparticles. This is the photocatalytic activity, methane blue dye degradation activity of magnesium oxide nanoparticles, and kinetic study of photocatalytic activity of magnesium oxide nanoparticle. This is the antioxidant activities using the magnesium oxide nanoparticles. So this is the inhibition percentage of antioxidant activity is the higher concentration. Next slide shows anti-cancer properties of magnesium oxide nanoparticles were analyzed using human melanoma cancer cell lines A375 via MTT assay, XTT assay, and NRU assay. This is the antimicrobial activity of synthesis of magnesium oxide nanoparticle. Antibacterial activity of the 4 bacterial strains. This A is the DMSO, B magnesium oxide nanoparticle, C is bark extract, the plant extract, D is the standard antibiotics. We used 4 bacteria. So next slide shows nanoformulation using bioactive molecules. Nanoformulation, this nanoparticle we are using for the nanoformulation, nanogels, dendrimer, polymer nanoparticles, liposomes, polymeric micelles, nanoemulsion, solid nipple nanoparticles. For using the bioactive compounds like quercetin, curcumin, resveratrol, like glucosinols. This work published recently in this chemical biological interaction. This is the encapsulated in various types of nanocarriers, targeted delivery of in the cancer cells. Phytochemicals combined with the various nanoparticles, nanocarriers, liposomes, nanocomposite, micelles, dendrimers, like these nanoparticles combined with the phytochemicals, loading. This is the drug delivery to target the cancer cells. These results was published in the current nanoscience. This figure shows curcumin liposome nanocarrier to enhance drug delivery potential. Curcumin with the liposome nanocarrier to transfer the mechanism of this is the mechanism, the drug delivery mechanism. This work was published in Seminars in Cancer Biology, 2022. This figure shows pharmacokinetics, absorption, distribution, metabolism, and excretion of personalized drugs of drug combined with nanoparticles. So this is the absorption, distribution, metabolism. So this is the advantage of like a nanoformulation. So this is the biomedical applications and also nanoformulation of nanoparticles. So next is application of nanotech in agriculture. So nanotechnology is using for various applications in the agriculture, like farmer using high-tech devices in the field, nanoparticles for precise delivery for fertilizer, nanomaterial-based diagnostic tools for early detection, plant diseases and pests, healthy growth and increased productivity, and nano tools used for precision breeding, monitoring and analyzing soil water contaminants, enhanced crop protection and reduced chemical usage, nanosensor for real-time monitoring of soil conditions. So this is the applications, various applications of nanotechnology in agriculture. So we have effect of nanoparticles in the in vitro grown Chinese cabbage seedlings. We checked different concentration, control and 10 mg, 50 mg and 500 mg. So 500 mg, effect of 500 mg higher concentration, the plant was stunted the growth, root and shoot. So low concentration increased only roots. High concentration decreased root and shoot. So this is the this graph shows shoot percentage, shoot length and root length, plus biomass and number of nickel content, total chlorophyll. Chlorophyll was decreased in the higher concentration. Biomass also decreased in the higher concentration. Nickel content increased in the higher concentration, root and shoots. This figure shows carotenoid content gradually decreased in the higher concentration. Like anthocyanin gradually increased in the higher concentration. Total proline gradually increased in the higher concentration. And sugar content gradually decreased in the higher concentration. MDA content gradually increased, root and shoot. And H2O content gradually increased in the shoot and root. So this is the this slide shows POD content gradually increased in the higher concentration, and also ROI's generation gradually increased in the higher concentration. The green fluorescence indicates the presence of ROIs in the roots. The higher concentration increased, fully increased. So lower concentration is gradually decreased. This is the high toxicity of the nanoparticle. So this slide shows the bioactive compound. Glucose. We did UHPLC. We analyzed the treated nanoparticle treated plants. So we analyzed glucosilin, cinegreen, various glucosinols we have identified. The level was higher in the treated plants. And the phenolic compound also in the total phenolic and flavonoid also higher in the treated plants. Next slide shows different concentration of nickel oxide nanoparticle in the mRNA expression levels. We analyzed antioxidant enzymes, GATE, POD, and GST, and the phenolic biosynthetic genes, POL, ANS, and PAP1, and aliphatic glucosinol biosynthesis, MYP28, MYP29, and the indolic glucosinol, MYP34, MYP51 transcription factor genes. The gene expression was in the higher concentration, CAT and POD, GST, POL, ANS, PAP1. Also, the expression was higher in the higher concentration. Like MYP28, MYP29, MYP34, MYP51 also higher in the higher concentration. This is the glucosinol genes. This is the phenolic compound related genes. So this figure shows nanopriming as a emerging seed priming technology for sustainable agriculture. So nanoparticle using for nanopriming, various nanoparticle using for the nanopriming. Nanopriming is an innovative seed priming technology that helps to improve seed germination, seed growth, and yield by providing resistance to various stresses in plants. Nanopriming is a considerably more effective method compared to all other seed priming methods. The salient features of nanoparticles in seed priming are to develop electron exchange and enhance the surface reaction capabilities associated with various components of plant cells and tissues. This slide shows influence of nanopriming on the primary and secondary metabolism in plants. After nanopriming, the changes of ROAS and primary metabolism and secondary metabolism also changed in this. Improved defense mechanism, stress tolerance, yield. This work was published in the Journal of Nanobiotechnology. Nanoparticle used as a elicitor. Biologically synthesized nanoparticles, silver nanoparticles, to enhance the phytochemicals, glucosinols, and phenolic compounds. The transcription levels and their biological activities in genetically transformed root cultures of Brassica repa. The concentration of silver and reactive oxygen species, MDA or hydrogen peroxide, were highly elevated in the AGNP silver nanoparticle elicitor hairy roots. Glucosinols such as glucolysin, glucoprasecanopin, cinegreen, proconitrine, gluconopin, 4-methoxyglucoprasin, and 4-hydroxyglucoprasin, glucoprasin, neoglucoprasin, and gluconostrine, and their transcripts MYP34, MYP51, MYP28, MYP29 were significantly enhanced in the AGNP elicitor hairy roots. Phenolic compounds like flavonols, hydrobenzoic, and hydrocinamic acids were highly enriched in the AGNP elicitor hairy roots. Total phenolic and flavonoid concentration and the transcripts of POL, G, and FLS were higher in the AGNP elicitor hairy roots than the non-elicitor hairy roots. Biological activity like antioxidant, antimicrobial, and anti-cancer activities were significantly higher in the silver nanoparticle elicited hairy roots than in the non-elicited hairy roots. The silver nanoparticle elicited carrier cultures offered an efficient and promising in vitro method to increase the production of health-promoting bioactive compounds, which may be useful nutraceutical and pharmaceutical industries. This work was published recently, Biosystem and Engineering, 2018. So finally, it's the conclusion of this talk. The field of nanobiotechnology holds immense promise for revolutioning various sectors from medicine to environmental conservation. The innovative amalgamation of nanotechnology and biotechnology has paved the way, groundbreaking advancements, enabling precise manipulation at the molecular level. The applications of nanobiotechnology are vast and transformative. In medicine, targeted drug delivery system promise personalized treatments with minimal side effects, while biosensors offer rapid and sensitive diagnostics tools. The environmental applications include pollution remediation and efficient energy protection. Thank you. Thank you for all. Thank you.
Hello. Hello.
Yeah, thank you. Thank you, ma'am.
Thank you so informative lecture. Sir,
Sorry.
No issue, sir. Finally, you have
Yeah.
conducted a very good lecture, sir. So now we will start this oral presentation. Sir, question answer session we will discuss after the oral presentation.
Okay.
We will call you, sir, at that time, sir.
Okay, ma'am. Okay, I'm in the online. Okay.
Thank you so much.
Thank you.
So for the oral presentation, we will start our section next 15 minutes. So you people just ready with the presentation and you have a time of only the 10 minutes for per presentation. Okay, so I'll call the name of the student. Okay, the presenters, Dr. Somali Sanyal, Miss Deepali Sharma, Miss Anushri Nair, Miss Shelly Agrawal, Miss Garima Yadav, and Miss Vrinda Devi, and Miss Archana, Mr. Patel, and the last one is Mr. Jeevan Patra. Okay, finally, one more invited talk with us, Dr. Pankaj Rai. Rai sir is there. So the Pankaj Rai sir, first of all, deliver his lectures. Then afterward, we will start the oral presentation. As the name I discussed here. Okay, start with the Somali one first. So Somali, first of all, Pankaj Rai will deliver the lecture. So welcome, sir. So first of all, I brief about the Dr. Pankaj Kumar Rai, sir. Currently he's a professor and head of the Department of Biotechnology in Vertiz University, Bareilly, Uttar Pradesh. Dr. Rai has over the 14 year of extensive research experience and the 11-year post-PhD in teaching and research. Dr. Rai is a leading expert in his field, and his research focused on the innovative approach to the sustainable bioenergy production with the particular emphasis on integrating dark and photo fermentation process of hydrogen production from the dairy and the agro waste. Dr. Rai holds a PhD in biotechnology from BHU and has gained numerous accolades, including the prestigious fellowship from UGC, CSIR, and MNRE. He has published a 76 indexed paper with a total impact factor 96, and his work has been cited 1,424 times and reflecting his significant contribution to the scientific community. As a dedicated academician, researcher, administrator, Dr. Rai brings a wealth of expertise to advance discussing on cutting edge research and innovation at the international level or the national level. So I would like to call our next speaker, Dr. Pankaj Rai, sir.
Thank you, Dr. Asaf. Good afternoon, everyone present over here. First of all, I beg sorry, and due to delay, I was engaged in some guest lectures, and now I am free now. So I first of all I would like to thank the organizing committee members of this conference, especially Dr. Vinay Dwivedi and my friend Dr. Manish Srivastava, who has provided me this platform to share my research experience with such a great gathering of learned people. And my title of the talk is Optimization of Metal Ions Concentration in Photohydrogen Production, Photobiohydrogen Production. So the persons present over here might be knowing about the hydrogen, because everyone is talking about global climate change, sustainable energy, sustainable development. So, and everyone is thinking that by 2050 we will become hydrogen economy. The world will become the hydrogen economy. Means that all the transportation, all the energy supplies will be dependent, completely dependent upon the hydrogen. So hydrogen production may be carried out by two methods, basically biological, and one is the non-biological. So non-biological methods are not so eco-friendly. You may say that they are very energy intensive. They require much more chemical treatment, and they are not so eco-friendly. So the option left with us is biological methods. Okay, why biological? Because they can be operated at ambient temperatures and pressure, and thus they are less energy intensive. They are obviously they are environment friendly, and they can utilize renewable energy resources like organic waste from kitchen, from a canteen, from agriculture. They could be utilized for the production of biohydrogen. Only the cons available with the biological, yeah, you may say that reported in biological methods is the efficiency compared to non-biological one is very less. So this is a diagram showing the different type of biological hydrogen production, like direct photolysis, indirect photolysis, dark fermentation, and photofermentation. In direct photolysis, the algae could produce hydrogen with the help of protons generated after the photolysis of H2O, which result into the utilization by hydrogenase, which produce hydrogen. And in direct photolysis, the cyanobacteria, which is also a kind of algae, utilizes the photosystem 1 and photosystem 2 to produce the hydrogen with the help of NIF complex, or you may say nitrogenase enzyme. In dark fermentation, it is the independent of light. That's why we call it dark fermentation. In this type of fermentation, the fermentive bacteria like E. coli, Enterobacter erogens, Clostridium, they utilize the organic compounds like glucose, sucrose, lactose to produce hydrogen. By fermenting these organic compounds with the help of hydrogenase enzyme, or you may say that FHL complex, they produce hydrogen. And the last one is the photofermentation, which is carried out by the photosynthetic bacteria like Rhodopseudomonas, Rhodobacteria, Rhodospecies, Rhodobacter species. In this, the volatile fatty acid present in the medium could be utilized to produce hydrogen with the help of nitrogenase enzyme. And I am more concerned about the hybrid system, which consists of dark fermentation as well as the photofermentation. Why I am concerned about this? Because it has the capacity to increase the efficiency or the rate or the yield of the hydrogen, which is the target of the today's world. So this hybrid system, anaerobic bacteria like Clostridium, Enterobacter, or mixed culture of Consortia could produce hydrogen with utilization of glucose, or you may say any simple sugar, or you may say that the organic waste present in the medium could be utilized to produce 4 molecules of hydrogen per molecule of glucose consumed. With concomitantly 2 molecules of acetic acid or lactic acid or butyric acid also produced during the dark fermentation, which is present after the fermentation and is always thrown out of the site. But I planned to use this spent medium in the photofermentation. Okay, so what we do in the photofermentation, the spent medium collected from the dark fermentation of the organic waste, we expose this spent medium, pre-treat this spent medium, and the culture like Rhodobacter sphaeroides, Rhodobacter polystris, or Rhodopseudomonas species is grown in this type of medium. And what we observed that around 8 moles of hydrogen could also be harnessed from this spent medium. So in totality, we may calculate that theoretically 12 moles of H2 production is possible with 1 molecule of glucose. But Right now, we are approaching only 7.5 moles of H2 per mole of glucose. This means that there is a lacuna in the system which could be overcome to produce 12 moles of H2. So I focus on the metal ions concentration in the spent medium because the metal ions has the capacity to regulate the microbial growth and activity of the microorganism as well as the enzymes involved in the fermentation process. So hydrogen production is also affected by the metal enzyme concentration or metal ions concentration because the enzymes involved in hydrogen production are hydrogenase and nitrogenase. Nitrogenase has the metal ion centers like nickel iron, vanadium nickel, vanadium iron, iron sulfurs, and FHL complex. They all require the metal ions, iron, nickel, magnesium. So my focus was to optimize the concentration of these metal ions in the spent medium for the photofermentation so that the photofermentation of this spent medium could be done effectively. Also, if the hydrogen yield is increased by using this metal ion concentration, the cost effectiveness, or you may say that the cost of the production of H2 may be lowered down. So it will pave the way for the commercialization of the hydrogen production by photosynthetic methods. So first of all, we should know the what are the factors which affect the metal ion concentration in the spent medium. So there are mainly two factors which affect that. The first one is the biological factors, and the second one is the environmental factors. In biological factors, microbial metabolism and growth affect metal ion utilization. Okay, and the second one is the organism specific metal ion preference impact concentration. Environmental factors are pH, temperature, and oxygen levels influence the metal ion availability, and contaminants and substrates can interact with metal ions. So all these four factors, you must say the two factors, especially biological factors, environmental factors, may play a key role in the availability of metal ions in the spent medium. To carry out this experiment, I utilized two type of organism. One organism is Enterobacter aerogens for dark fermentation, and the second one organism was the Rhodopseudomonas BHO01, which was isolated by me only during my doctorate. It was used as a photosynthetic organism for the photofermentation of the spent medium. And the feedstock utilized was the cheese weight, 10 gram lactose per liter. That was the concentration of the feedstock. And the after the dark fermentation of this spent medium of this cheese whey, I obtained the spent medium and supplement this spent medium with nickel as a nickel chloride range from 0 to 8 micromole per liter, and iron as FeSO4.7H2O from 0 to 100 micromole per liter, magnesium ion as MgSO4.7H2O and 0 to 15 millimole per liter. It's not moving. Okay. After dark fermentation, the spent medium was analyzed, and it was found that lactose utilization was 79%, acetic acid production was 1900 mg per liter, and butyric acid percentage was concentration was 537.2 mg per liter, and the total COD reduction was 20%. Okay, it indicates that the dark fermentation was successfully implemented in the cheese wastewater, and I supplement this spent medium with nickel, iron, or magnesium. And this is the table which shows the effect of nickel, iron, and magnesium on S2 production by Rhodopseudomonas BHO01, and the reactor volume was 100 ml, and pH temperature was adjusted as per the requirement of the culture. Like temperature was set at 34 degrees centigrade, pH was set to 6.8, 200 rpm was maintained, and the time for fermentation was 96 hours. And I conclude that from the results that the concentration of nickel in the range of 4 microliter per liter was the more efficient for the S2 production, whereas nickel was found to be 60 micromole per liter and the magnesium was found to be 6 millimole per liter. Concentration was the best suited for the S2 production. So I figure out that the ratio of 1:15 is to 1.5 for nickel, iron, and magnesium is more suited for supplementing the spent medium. Okay. So the effect of nickel on H2 production, rhodosodium on BHO01 strand, reactor volume 100 ml, temperature 34 degrees centigrade, pH 6.8, and the RPM was set to 200 RPM, and the fermentation time was 96 hours. I found that in comparison to control, the sets with the 4 micromole per liter of nickel is producing 144 ml of hydrogen with the rate of 25.83 mole H2 per mole per liter spent medium, and the final yield was 58.06, which is a very impressive. It is almost 1.6 fold of the in comparison to control. It indicates that the supplementation of nickel is successful, and we may utilize this concentration for the supplementing the medium. This is the slide which is showing the effect of iron on the H2 production, and I conclude that utilization of 60 micromole per liter of iron will result into the almost 1.5 fold of hydrogen yield. Like in control, it was produced only 91, but in 60 micromole per liter sets, hydrogen was produced 139 ml. Rate was very much high in 60 micromole per liter sets. It was 24.16 in as compared to 17.5 ml of control, and the yield was 56.04, and in control it was only 36.69. And this is the effect of magnesium on S2 production by rhodosodium BHO01 strain, and the sets having 6 micromole per liter of magnesium supplementation produce highest S2 yield rate, or you may say the volumetric yield. Volumetric yield was 111 ml, rate was 21.66, and yield was 44.35. It is almost 1.2 fold. So you may see from the result that the magnesium ion has 1.2 fold effect, iron has the 1.5 fold effect, and nickel has the 1.656 fold impact. So in totality, we may say that the nickel has the highest impact on the H2 production rate and yield, and the magnesium has the least impact on the hydrogen production rate and yield. But all the supplementers, all these metal ions is beneficial. So you may conclude that nickel, iron, and magnesium at 4 micromole per liter, 60 micromole per liter, and 6 millimole per liter respectively were stimulated to H2 production, and magnesium did not affect H2 production significantly compared to nickel or iron, as 6 millimole magnesium sets produce only 120 ml H2, a value only 16% over the control. Magnesium is required for proper growth of the organism, as it lacks culminates in weakening of bacterial cells and the loss of colony characteristics. So the overall potential of trace elements in enhancing photobiohydrogen production from spent medium could be arranged as nickel is the most essential, followed by iron, and the last one is the magnesium ion. The study shows that the nutrient poor and VFA-rich spent medium needs supplementation of nutrient elements to qualify as the effective feedstock for hydrogen production during its subsequent photo fermentation. And this study is published in the International Journal of Hydrogen Energy, which is a reputed journal in the field of hydrogen, and it having the impact factor of 5.2. So thank you from my side. I think this is the brief overview of my research. Till now I have reported.
Thank you very much, sir, for the very informative lecture and the presentation over the metal ion and their applications. Now we are open for the panel discussion. Maybe Dr. Sushma Chauhan and Dr. Muthu and Dr. Vinay Dubedi, sir, is
Thank you,
there,
Dr.
and the
Pankaj.
Pankaj
Thank
Rai
you
is
very
there.
much.
Yes, ma'am.
Thank you, sir. Thank you. Is there any query?
I would like to call everyone, sir, please, if you have any query, any doubt related to the topics and want to know anything about the new innovative idea about from the Pankaj Rai, sir, you can ask.
Afternoon, Vinay, sir.
Good afternoon, sir. Good afternoon.
Nice to see you, sir.
Yeah.
Manish, sir.
Thank you, Dr. Pankaj.
Afternoon, sir. Afternoon. I think there is no query.
Sir, Dr. Susma here. I would like
Hello,
to
ma'am.
ask, hello, sir. Pankaj sir,
yes.
can you hear me?
Yes, very much. Please.
Thank you so much, sir. Sir, I had like a couple of questions because one of my scholar is working in biohydrogen production via metabolic engineering in E. coli. So, sir,
That's great.
as you thank you so much, sir. So you had done very good work on hydrogen production, I must say, and I have a small doubt that how you are capturing the hydrogen gas after the detection. Is there any because we have to think about that part also after
Yes,
the production part,
yes.
right?
We have to collect it very cautiously because it is highly inflammable.
Yeah, that's what
Yes,
I was.
yes.
How you are
I
like
collect
collecting it?
actually there is a very famous method for collecting the hydrogen, that is the water displacement method.
Okay.
You might be knowing that. And it
Yes,
is
sir.
reported by various authors, and I also reported in my papers that the what we do in the water displacement method, we use the solution of 30% of KOH, or you may say NaOH. Okay, and we put it in a jar, larger jar having volume of 2 liter or 3 liter. Then we use a measuring cylinder, transparent. You may use that perplex or glass measuring cylinder. Okay, and we fill the glass up to the top of the jar, and then inverted it, the jar inverted, and put it inside the KO 30% of KH. Okay.
Okay.
After that, I will send you the video also. I have the video. Okay.
Yes, sir. Yes, sir. It would be helpful for me
Yes.
actually.
And after that, what we do, we put that reactor outcome. The gas is coming from the
Outlet
reactor.
that
Na?
on the
We use the silicon tube to channel this produced hydrogen to the measuring cylinder. Okay. And this air coming out from the reactor contains H2 plus CO2 both. Okay. So when this CO2 and H2 comes inside the jar having the 30% of KOH or NOH, what it occurs, the CO2 is dissolved in the NOH or KOH, producing carbonic acid. Okay. So only pure H2 is there. And it will displace the water present over the measuring cylinder, and the water will going downside. Like the water comes down 1 ml, 2 ml, 3 ml, 4 ml. Okay. The water will be displaced due to the pressure of the gas. Okay. And we may calculate the moles also, like 22.4 mole, 22.4 liter of hydrogen is equal to 1 mole. Okay. So we may convert it into the moles also.
Moles, yeah.
Yes.
Dr. Pankaj, is there any purification process is required that is collected at the above place or at the surface of the water for this hydrogen gas that is obtained from this Pseudomonas?
Automatically CO2 will be dissolved inside the NOH or KOH.
Okay. Thank you so much. Thank you so much for your deliberation.
Anurag sir, good afternoon. How are you, sir? I think you have a very good team, sir. You people are doing great. I'm really happy for you people.
आपका talk सुना, it was very
Due to time constraints, sir, I am not able to discuss all very much. But
that we understand.
Yes, sir.
So we'll try next time in physical mode. So
Yes,
we'll
sir.
have
That will be great, sir. That will be great.
Yes. Sure.
Because in online talks, na, sir, we have to engage here and there on meeting, in classes and lectures, all this. But when we are there physically in the conference, na, it we all already focused to the conference only, not bothered about all the things.
Exactly.
Yes, sir. So I think कोई और questions नहीं है.
Dr. Pankaj, I will contact you later regarding the same.
Yes.
Yes, sir. Thank you so much, sir.
It's a pleasure, ma'am. Okay. Thank you all for inviting me, for sharing this platform. Thank you.
Thank you, sir. Thank you for joining the session and deliver the talk. All the invitees speaker are very thankful to you all. Now I would like to start the oral presentation session. So one by one we call to all the participants. So first candidate is Dr. Somali Chauhan. Dr. Somali Sanyal. Are you there? Dr. Somali?
Ma'am, please announce if they are in attendance, so they can just raise the hand, so we can just promote.
Okay. Dr. Somali Sanyal, first oral presentation is yours.
Please raise your hand, ma'am.
I think she's not here.
Okay. Next is Miss Deepali Sharma.
Whoever the paper presenter, just raise the hand so we can promote you in the panelist.
Okay, all the oral presenter, please are requested, raise your hand so we will make you a panelist. Yeah, 2 panelists. Three attendees. Sani, Jeevan.
Yeah, I just send you the request. Just join, please.
All the presenters are requested to complete your presentation within a 10 minutes. And afterwards, we will call the next participants. Okay, so please constrain the time frame. So the first presenter is Miss Deepali Sharma, Assistant Professor, Shri Guru Gobind Singh Technitary University, Gurgaon. So ma'am, please share your screen.
Ma'am, I think Dipali is not here. Just check the panelist name and just.
Next is Miss Anushri Nair. Anushri S. Nair.
Yes, ma'am.
Yes, connect yourself.
Ma'am, in panelist, there is Anushri, and then Garima, then Jeevan, then Sally. 1, 2, and 3, 4, 5 people are here.
Okay, so accordingly, whoever is present, so we will call one by one, sir. Okay, no issue.
Ma'am, is it visible?
Yes, it's visible.
Okay.
So let's start. Make it a full screen.
Yes,
Anushri. Also turn on your camera.
Yes, ma'am. Ma'am, is it visible now?
Now it's visible.
Good afternoon to everyone. I am Anushri S. Nair from Satyasai College, Assistant Professor, Biotechnology. And I'm here to present a paper on the topic biodegradation of toxic chemical pesticides by inoculum of nitrogen-fixing bacteria and its synergistic effect on soil fertility. First of all, we'll discuss about the introduction of nitrogen-fixing bacteria in soil. In the realm of agricultural science, a revolution is taking place, harnessing the power of nitrogen-fixing bacteria to enhance soil fertility and combat the negative effect of toxic chemical pesticides. This groundbreaking approach employs a synergistic biodegradation ability of Rhizobium japonicum and Azorhizobium colinodens, offering a sustainable and eco-friendly solution to boost crop productivity while safeguarding the environment. So here are the negative importance of soil fertility. In order to achieve a sustainable and productive agriculture, maintaining soil fertility is an utmost important. Nitrogen-fixing bacteria play a crucial role in enhancing soil fertility by converting atmospheric nitrogen into a usable form for plants. By harnessing the power of these bacteria, we can reduce the reliance on chemical fertilizer and promote a healthier and more eco-friendly approach to farming. These are the negative impacts of toxic chemical pesticides, where we can see that the pesticides which are used in our farms, in our agriculture field, which impact health, it causes problems in diseases in pregnant mother and children, drains economies, decreases biodiversity, and it has negative impact on soil, water, and air as well. Here, we will discuss about the synergistic biodegradation combining nitrogen-fixing bacteria and pesticide breakdown. In our research, we used two bacteria strain, that is Rhizobium japonicum and Azorhizobium collinodens. We found out that not only these bacteria can integrate nitrogen fixing in the soil, and they can also degrade the pesticides which we have used as organochlorines and organophosphate. The organochlorines and organophosphate, when we mixed with these bacteria, found out they were degraded into simpler substances. So by combining the nitrogen fixing bacteria and utilizing the ability to break down the pesticides, we can create a sustainable solution for agriculture. This synergistic process not only reduces the environmental impact of the pesticide, but also enriches the soil with essential nutrients, promoting healthy plant growth and increasing crop yields. And in case of organochlorines, we found both bacteria exhibiting enzymatic actions such as dechlorination process and the presence of dehalogenation, which contribute to the breakdown of the compounds. And for organophosphate, the enzymatic machinery, particularly organophosphorus dehydrolase and phosphotriesterase, these enzymes played a very vital role in cleaving phosphorus ester bonds, facilitating pesticide degradation. Now, the objective of our research was production of potential or biofertilizer, degradation of chemical pesticides, antagonistic activity against some pathogenic bacteria and fungi. Not only this, these bacteria also provided plant growth promoting hormones, which were an essential nutrients for the plant growth and increase in yield productivity. Methodologies were isolation of bacteria from plants. We used plants as Bania rostrata and soybean for the isolation of bacteria from its nodules. Next, we did morphological and biochemical test for characterization and identification of microorganisms. Here we did barcoding also, analysis of enzyme activity by HPLC, then the estimation of nitrogen fixation by Geldahl's method, screening of potential pesticide degrading bacteria. Last, we tried pot trials using we used guar gum as a carrier for the biofertilizer. These are our test results, where we did for both isolates of Rhizobium japonicum and Azorhizobium collinodens for the biochemical test. We did ultraviolet treatment, effect of metal test, citrate utilization, bromothiamyl blue, urease test, starch hydrolysis, utilization of carbon source, indole acetic acid production, and catalase test. These are the results. And these are the results for the rest of the biochemical tests that we did. This is the HPLC report, where we found out that the organophosphates and organochlorines were degraded into simpler substance by our Rhizobium japonicum and Azorhizobium collinodens. Biodegradation of malathion where media constituent, we used YEMA without any nutrients, where we introduced YEMA without pesticides, and we got a mean value of 324. YEMA with pesticides, we got 28 colonies of T2 and T1 are 31.6. Yama without nutrient, that is mannitol and yeast extract plus pesticides, we got around 411 and 340 mean value. T1 is the Rhizobium japonicum and T2 is the Azorhizobium colinodens. These are the graph which we obtained. And we also did a combination of both T1 and T2. We found out that without nutrients and using the pesticides, the bacterial isolates grown where a number of colonies that grow were high as compared to individual bacterial isolates. These are the pot trials that we got from our pot trials. T1 broth, it yielded 150 to plus minus 3 grams. And these are the results. So the conclusion is that the potential of nitrogen fixing bacteria in enhancing soil fertility and reducing the use of chemical pesticide is significant. By harnessing the power of synergistic biodegradation, we can promote sustainable agriculture practice and ensure a healthier and more environmental friendly future for our planets. Thank you.
Thank you, Anushree. Am I audible to you, Anushree?
Yes, ma'am.
Anushree, one question from my side.
Yes,
you have mentioned in methodology bacterial isolation. You have mentioned in methodology bacterial isolation. So which plant part you have used for the bacteria isolation?
Ma'am, we have used root nodules and for Sesbania.
Your voice is not coming, Anushree.
Ma'am, is it audible now?
Now it is.
Ma'am, is it audible? Ma'am, we used root nodules and plant nodules.
Okay. You have mentioned the name of bacterial species, right?
Yes, ma'am.
So have you done or go for the molecular characterization?
Yes, ma'am. We
Because
did
you
a
have not.
Yes, ma'am. I haven't attached the result of barcoding. Sorry, I just missed that. But yes, we did a barcoding of Rhizobium japonicum and Azorhizobium colinodens and did the biochemical test also. We confirmed through the barcoding, ma'am.
No, this is my concern. A biochemical test is not sufficient. If you
Yes,
have
ma'am.
done for the
Biochemical test is not
barcoding or the
sufficient, ma'am.
molecular characterization,
We went.
which is not
Yes,
in
ma'am. We a
it is
result,
not in
ma'am.
your
Sorry.
presentation.
Yes, ma'am.
It is not mentioned in your presentation, so you must include this for
Yes, ma'am.
next
Sorry,
time.
ma'am. I just missed that out. Okay.
It is okay. Thank you.
Thank
Can
you,
I
ma'am.
ask one question, ma'am, if you allow?
Yes, ma'am.
Yes, ma'am.
Thank you, Neha, ma'am. Thank you, Asha, ma'am. Ms. Anushree, throughout your slides, I didn't see any control sample. So how you will?
Ma'am,
Yeah.
we did a control samples. Lengthy, ma'am. So we took a control samples of for just bacteria of using pesticides and all. Sorry, ma'am. Sorry. We used a control, ma'am, for Rhizobium japonicum and Azorhizobium. We did a control test without pesticides, and with pesticides we included both as a control, and we found out that the plant just the pot trials where it the normally we just included that bacterial isolates, it was good enough, but when we used pesticides for a degradation, the plant growth yield was high, ma'am.
But in your slide, you have not mentioned. So how is how you will justify that? Because without control, you cannot justify, and you cannot say that it would be lengthy. So you didn't. You cannot say that you missed it out, right? Any control,
No,
like
ma'am.
positive or negative control, you should have mentioned in your slide.
Yes, ma'am. Yes, ma'am.
So next time, take care of those things, okay?
Yes, ma'am. Of course, ma'am. It's my first time for international conference, so that's why I just
it's fine. Yeah, okay. Thank you, ma'am.
Thank you. Now, next presenter is Miss Shelly Agrawal. Miss Shelly, are you there?
Yes, ma'am.
Please upload
Yes,
your
yes, ma'am.
presentation.
Yes, ma'am. Just a minute. Hmm. Is it visible, ma'am?
Yes, it is visible, Sally.
Yes,
Please
Shelly.
start your presentation.
Yeah. So good afternoon, everyone. Myself, Shaili Agarwal. I'm from Amity University, Noida, a PhD scholar working under the guidance of Dr. Alok Kumar Singh. Today, I'm here to present my poster on evaluating the combinatorial effect of glycopeptides and beta-lactams against antium. So first, what are antiums? Antiums are environmental pathogens, non-tuberculous mycobacteriums, bacterium,
Sally, just make it full screen better. Because it is in form of poster and it is not visible to us.
Yes, ma'am.
Make it in full screen.
Full screen.
Shall just press button F5 on top here. Press F5.
Shift.
Anyway, Shali, you can just bring your title on the screen and accordingly you can scroll down. Whenever you reach the objectives, you just scroll down so that the audiences can view.
Is it now? Is it visible, sir? I mean,
Yeah, we can see your abstract, we can see your title.
yes, sir. Yes, sir. Um, Antiums. So, antiums, non-tuberculous mycobacterium, they're environmental pathogens and ubiquitous in nature. They cause multiple infections like pulmonary lung infection, skin infections. Uh, uh, Antiums share the same genera as of Mycobacterium tuberculosis, but the notable difference between Antium and MTB, Mycobacterium tuberculosis, is the tackling of Antiums. Like they are drug resistant to those antibiotics that we use for treatment of TB also. The first line drugs used for TB, like isoniazid, rifampicin, and pyrazinamide, they are drug resistant for these drugs also. That's why they are very tough to handle. And currently, there is no any clinically drug present for the treatment of antiums. So, this is a very challenge. This is very challenging antium to treat them. Uh, because antiums are intrinsically drug resistant. This is because of the structure of cell wall. Their cell wall is highly lipid. Their cell wall is rich in lipid rich. That's why, and this lipid rich cell wall confers them intrinsic drug resistance. That's why that provide the low probability of cell wall and make these antiums drug resistant. Historically, there is evidence present that glycopeptides and beta-lactams show synergy in action, but this strategy has not been used against antium yet. So here I am repurposing the glycopeptides and beta-lactams against antium for the treatment of antiums, the combination of glycopeptides and beta-lactams. For this, I have two objectives. First objective is to determine MIC for different glycopeptides and beta-lactams against different strains of antiums. Second, to determine the FICI and synergism between them. FICI fractional inhibitory concentration index or signifies the value of synergism between two drugs, like whether these two drugs are for this methodology. First, MIC. To determine the MIC,
I prepared drug stocks for various concentrations and incubate the drug solution with the bacterial. Same for FICI, but in case of FICI, I prepared drug stocks for both drug solutions and combined them. Then I noted down the MIC values, like I observed the reduction in MIC values after that. So here are my results. First MIC, first table is for MIC simple. MIC for I have choose some beta lactams and glycopeptides against entom strains. Like I have used Mycobacterium abscessus kiloni for tutum. Next, I did FICI. I find FICI. So here we can clearly see, like if I talk about first table for FICI. So when I was using like GL1 glycopeptide 1 alone, then its MIC was 16, and same for beta lactam 1. When it was alone, its MIC was 16. But when I combine them, glycopeptide with beta lactam, like glycopeptide 1 with beta lactam 1, then its MIC reduced to 4. Same goes for beta lactam 1, and some IC reduced to 4. And we can see this reduction in MIC value in every combination: beta lactam 1, beta lactam 2, beta lactam 3 with glycopeptide 1 and glycopeptide 2. In all combinations, there is a significant reduction in MIC values. Plus, these combinations, all these combinations are synergistic. The FICI value is 0.5 or below than 0.5. That means these drugs are working in a synergism manner, supporting each other in the reduction of MIC. Like we can see 0.5, 0.2. In all combinations, FICI value was 0.5 or below 0.5. So, conclusion, we can clearly see that I repurposed glycopeptides in combination with beta lactams for entom treatment and found all combinations were synergistic. So these results suggest that the combination of beta lactam and glycopeptide is significantly effective. And if the in vivo animal data also support this, then it can be clinically tested to examine its potential to reduce the long chemotherapy associated with entom infections. Currently, I have done this in vitro. Afterwards, I will do this for in vivo also. Yeah. Thank you, everyone.
Okay, Shelly, thank you. Wait some time. Sir, you can ask the question, please.
Yeah, Shelly, am I audible? Yeah, my question is that you have conducted the MIC
Yes, sir.
experiments against certain
Yes,
bacteria.
sir.
Okay. So did you conduct this experiment while calculating the CFU of bacteria?
Yes, sir.
So how did you calculate the CFU of bacteria?
No, sorry. No, not calculated bacteria on a 96 simply different concentrations of drug stocks and better culture of a specific OD. So simply visibly it was visible. Visibly I can see the growth on the bottom of plate. What is MIC? In which well the growth is not present. So that is the MIC of that drug.
I understood the interpretation of MIC. My question,
Means that
Shall you please listen to me? My question is that while conducting the MIC experiment, you had added certain bacteria, okay? And then you have gone through the study whether they are being inhibited by your compound or not. My question is, what was the CFU of bacteria, initial bacteria against which you have studied the CFU experiment? Because let's say one of the bacteria, I don't
sir,
remember
I
the name of
used
the bacteria. I don't remember, but there must be some infectious dose of that bacteria. So
yes.
if the infectious dose is, let's say, is 1 million cells, so at least 1 million cells should be taken into account for calculation of your MIC. So do you have done this?
Yes, sir. Yes, sir. I use 0.30 for the dilutions. Specific 0.3075.
And similar experiments were done for other bacteria as well.
Yes, sir. Same OD for all bacteria.
All right.
Okay, so the next presenter is Miss Dipali Sharma. Dipali, share your
Good afternoon, ma'am. Am I audible?
Okay.
Yes, Dipali, you are audible.
Okay, I
Please
just share
about
my.
bite the time constraint, okay? Only 10
Sure,
minutes.
ma'am.
So, Dipali, is there any problem?
Yeah,
What is it?
yes, ma'am. The screen is not, actually, the share screen option is not being selected by my cursor.
Why you are in the panelist, na? So you can
Yeah,
share
ma'am. I
your
am
screen.
in the panel.
Yes, just go to the bottom. There is a green arrow upright arrow is there. That is the share screen option was there. Just share your screen.
Yes, ma'am. I'm clicking on the share screen button, but it's not responding.
Okay, so we are moving forward. We are calling our next presenter, Miss Garima Yadav. Miss Garima, are you there?
Yes, ma'am.
Huh. Garima, start your presentation.
I'm audible, ma'am.
Yes, sir, you are audible, Garima.
Good afternoon, everyone. I'm Garima, PhD scholar under the guidance of Dr. Jyoti Mathur, and my review topic is waste-mediated
Are
nanoseed
Garima?
priming. Yes,
Hello.
ma'am.
Garima,
Yes,
your
ma'am.
presentation is not visible. You have upload your screen or presentation or not? You share your presentation, no?
मैम शेयर किया है मैंने.
No, it's not visible.
1 minute.
Yeah, now you have started. Double click to enter the full screen mode. Yeah, now
Yes,
it's
ma'am.
visible. Yeah, come to the first slide, first of all.
Yes, ma'am. Good afternoon, everyone. Today I'm here to present my review paper, Waste-mediated nanoseed priming, an emerging technique for improving plant growth and
Garima,
productivity.
hold on, hold on. Yeah. Your first slide is important.
Yes, ma'am.
Now you can start.
Okay, ma'am. Okay. In recent years, the application of green chemistry has become concomitant with advances in the synthesis of nanomaterials. Environmental sustainability is now one of the major design criteria for the synthesis of nanomaterial and their application. Hence, it is evident that synthesis procedures involve waste recycling, use room temperature aqueous solution and reduce uses of other toxic chemicals for synthesis of products, and most acceptable from the point of sustainability. In the European Union, approximately 89 million tons of food waste is generated, and this value is expected to increase 40-fold in coming years. The Food and Agriculture Organization estimates that around 40% of food produced in India is wasted. The study revealed 1 minute. The study reveals the essential peels and other constraints on vegetable and fruit common use. For example, the peels of avocados, grapes, lemon, orange, pomegranate, jackfruits, and mangoes contain 50% higher phenolic concentration as compared to fruit pulp. The fruit and vegetable waste can be utilized to extract as well as obtain bioactive compound that can be used in agriculture industries for plant growth promotion and to increase productivity. Nanoseed priming is a combination of nanoparticle treatment and a seed dressing technique. It is an innovative seed priming technology that helps to improve seed germination, seed growth, and yield by providing resistance to various stresses in plants. Now, the waste mediated green synthesized nanoparticle, we're taking like a peels from Musa paradisica, Carica papaya, Citrus cyanescin, Lycopersicum esculatum, Vitis vinifera, Punica gratum. After that, the seed priming through the biogenic nanoparticle, it helps the biogenic nanoparticle have to reduce using pesticides and chemical fertilizer. The nanoseed priming increases plant growth and crop productivity, like as compared to the nanoseed priming and normal growing plant. The nanoseed priming has a great effect that like a plant height, root length, shoot length, pod per plant, and quantity of grains, and the nutritional quality of grains like macro and micronutrients and mineral content in the grain. After that, in this figure, we represent the nanotechnology waste mediated nanoseed priming for sustainable agriculture. In approach of nanoseed priming that increase seed quality, seedling vigor, yield enhancement, and impart toleric and abiotic stress. The seed storage for the seed protection over time, the plant establishment, improved seed germination, plant growth. The plant resistance also abiotic and biotic stress. Food production and quality as increases. Sustainability use, reduce the pesticides. The finding of seed last research the Irico sativa treated silica nanoparticles exhibited increased growth characteristics and a germination rate up to 95% comparison to the control, and also see the fungicidal efficacy of biogenic silica nanoparticle that are forming from sugarcane baggage, and NPs against fusarium wilt was also evaluated. But study by Yasmin demonstrates seed priming with iron and copper nanoparticle improved wheat plant characteristics like spike, grain per spike, and great weight. And after that, we are also platinum nanoparticles increase the pea production in this. And the last highlighted benefits priming watermelon seed for biogenic silver nanoparticle, resulting in the significant boost in plant development and metabolic activity. In this figure represents mainly we use the vegetable feeds after the nanoparticles and the seed priming with nanoparticles. The process helped to maintain the ionic balance, boost antioxidant enzyme activity, reduce oxidative damage, and ultimately enhance stress resilience. And the other parameters reduce ROS and oxidative damage, regulated photosynthetic attributes, regulated antioxidant enzyme, produce secondary metabolite, maintain ionic homeostasis, the abiotic stress tolerance, and the crop growth enhancement using by the nanoseed priming. In this figure, in this A figure, effects of nanoseed during germination, and the B represents the impact of ROS level on seed nanopriming, and the C diagram represents that the potential consequences from both biotic and abiotic stress. These strategies approach hold promise for mitigating adverse effect on seed aging, providing viable means to enhance germination, indices, and seed viability under stress-reduced condition, boosting the production and quality of the crop. Similarly, Rahana et al. 2020 represented with the platinum nanoparticle to boost the crop. And after that, the Acharya et al. 2020 highlighted benefit watermelon seeds with biogenic silver nanoparticle resulting significant boost in plant development and metabolic activity. Nanoseed priming helps to increase high yield, increase nutrition quality, protect from biotic and
Garima,
abiotic
last
stress.
1 minute you have.
After that, we also yeah. After we also tried machine learning and AI for nanoseed priming seeds, infrared spectroscopy, and the data mining and curation X-ray images benefit for application in artificial intelligence and machine learning by improving utilizing profiling for heat, moisture content, seed priming technology. AI and ML may be better complement each other. And the diagram shows that after nano priming, AI and ML methods used to predict germination, vigor, and yield of the nano-primed seed. And the conclusion, the waste-mediated nanoseed priming represents a suitable, innovative approach in culture utilizing waste material enhanced seed. The application of nanoseed priming demonstrated positive impact on plant growth, offering healthier and more robust throughout life cycle. This resulted in grain productivity, suggesting it's to address global demand, crop yield, meet food security challenges. The positive outcome and productivity translate the economic benefit for farmer, potentially reducing the need for excessive agriculture inputs. And the future agriculture solution highlight potential for novel eco-friendly techniques to address the changing and feeding growing global population. I have
I have one question here. Am I audible?
Yeah, sir, you are audible. Garima, are you there?
Yes, ma'am.
Ha. Sir is asking the question, please.
I have
Yes,
one
ma'am.
question.
Yes, sir.
I'm the seed with nano suspension. And
Yes,
second
sir.
one is what is the underlying mechanism behind the stress tolerance that has been developed because of the priming?
Hello, sir.
Did you get my question?
No, sir, please pardon.
How did you prime the seed with nano suspension?
Sir, we take a different concentration. In my research work, sir, trials are going on and the
No.
different
What I am asking, how did you prime the seed? What is the procedure to prime the seed with nanoparticles? Nano
Sir,
suspension.
sir, I make a suspension and different concentration, and
Right.
we put a seed on different concentration and check the which concentration is more which concentration is more
For how long you have
effective.
kept the seeds in the nano suspension, then you must have re-dried it, and then you have put it up for the germination. That is what I'm asking. So what is the procedure of seed priming, not the germination? Well, how did you do before germination? How did you prime the seed?
So we put a suspension, no?
You
In
have
the
put up into
suspension,
the
sir.
suspension. For how long you have kept there?
Yes, sir. Hello. Hello.
How long you kept the seeds in the suspension for priming? This is what I am asking.
Sir,
After priming, you must have
sir,
kept the
I am
seed for the
taking.
germination, na?
Yes, sir.
So that is what my question
Sir,
is.
हमने
How did you
different
prime the seed? You must have taken the solution of different
Sorry, sir.
concentration.
Yes,
That is
sir.
true. But how did you primed? How you have coated the seeds with the nanoparticles? Then you have put up for the germination. That is what my question
Yes,
is.
sir.
Are you getting my point?
सर हमने आवर्स का आवर्स डिफरेंट आवर्स पे देखा है सर डिप करने के बाद
And then you kept
different
those and then you kept those seeds for germination under the different stress conditions.
Yes, sir.
ठीक है. Okay, fine.
Okay, thank you, sir.
Garima, I have one more question in addition to Professor. Beta, this is your own work or you are presenting the review article by collecting the articles? Because I didn't see any data from your side, but just the pictorial things and the writing part. So have you started experiment things or you are just doing the review literature?
Sir, ma'am, I write firstly review literature and my lab experiment were going on. That's why I'm not put up pictures in the slide.
Okay, so Beta, there is a need to understand things to present in the conference. Okay, you cannot present just reviewing someone's articles or things in the conference until and unless you don't have your own partial data. Okay, conference doesn't ask you to present your complete data set, but at least you should have initiated some of the things by your own, right? So next time be careful on that.
This is my
Yeah.
review, ma'am.
No, beta, this is not your review. You are reading for your clarification to finding the gap, okay? It's not your review. Even the words
Okay,
are
ma'am.
not yours. That's what I'm trying to say, okay?
Okay, ma'am. Okay.
Yeah, so try to understand those things, huh?
Okay.
Thank you.
Thank you.
Yeah.
Okay, the next presenter is Mr. Jeevan Patra. Jeevan Patra. Yeah, Jeevan, you are not audible.
Now it's audible.
Yeah, speak loudly.
No, now it's audible, ma'am.
Yes, now you are audible.
I hope the screen is visible. Will I proceed ahead?
Yes, Jeevan, your screen is visible. You can proceed.
Thank you. Now,
Also, turn on your camera.
my camera is switched on, ma'am.
Sure, okay. Now you are visible. Good.
Now, a very good day to all. In this presentation, I will be mostly focusing on the mostly remarkable hotspot target candidate on falciparum-16 proteases, which is been a crucial barrier in defending different plasmodium strains and resistance across the different epidemic across our global. Proceeding ahead with my studies. So although we have plethora of therapeutic interventions and drug targets along with some vector to mitigate and prophylaxis with all plasmodium strains, still why there is a research gap and what are the unmet needs that needs to be accomplished and fulfilled. To answer and address with these research questions, I proceeding ahead with my with this presentation. Mostly this is an overview of the research background of the current stats of the plasmodium affected victims and the strains all across the global. And although we have plethora of drug targets candidates which has already been biochemically and genomically validated, still there is also a need for searching of unexplored or rarely explored drug targets in finding out putative promising therapeutic clinical like candidates. So although we have many kind of drug targets, but why falciparum-16 proteases is a keen interest for all the scientific community in the current era, it's in the current time. So mostly falciparum-16 proteases inhibit in the formation of free amino acids These free amino acids are responsible for parasite survivability and osmotic balance of the parasite, which has been infected at the intraerythrocyte stage in the blood. So if we inhibit these oligopeptides, so there won't be formation of free amino acids. Ultimately, there will be a survivability, mortality index will increase of the Plasmodium infected RBC. So although since past 2 decades, we have plethora of publications, patents reported based on the falciparum drug targets, but the major research question is still yet none of the potent molecule which also been reported in a nanomolar inhibition, but still unable to reach in the clinical pipeline. Due to not reaching to the clinical stages or in even microdosing stages, the reasons are poor pharmacokinetic, selectivity in terms of off-target engagement with highly homologous human cathepsins, where a drug is much more susceptible and to target Plasmodium, as also with human cathepsins. Means these human cathepsins are highly homologous. To address all these research questions, our research group have identified 2 molecules which has been confirmed through the in vitro studies and been published in past 3 years earlier. Mostly these 2 target I abbreviated as NM12 and NM15, which are having a micromolar of inhibition range. So our main intention is to optimize these 2 molecules in terms of traditional medicinal chemist and in a ring decoration. So that molecule, these 2 identified molecule, which is having a 2 unexplored scaffold, indole 2-carboxamide and tetrahydroquinoline. These 2 scaffold have not yet been reported since past 2 decades. So we identified these 2 molecule to optimize, which can inhibit the falciparum. Falciparum have a dual homologous, falciparum 2 and 3, which is a cysteine proteases, and these are the global conserved residues, and human cathepsin K, HCAT-K, I annotated here. So mostly human cathepsin K is highly homologous with Plasmodium. So our intention is to make such kind of molecules which can only target falciparum, means with Plasmodium, but not with the human cathepsin K. So to confirm it that we mostly work on the molecular dynamic studies. Mostly this presentation, whole research outputs are based on the informatics approaches, bioinformatics. So we confirm through the dynamics how the orthosteric groove is active with the co-crystal ligand, which has been reported. And further, we had a sequence-based alignment of falciparum with cathepsin K. Although we have total 11 cathepsins K, and these are the sequence alignment, and color is been annotated based on the residue's nature, and there is total 4 subtypes. And here I tabulated all the human cathepsins K subtypes and isoforms. So among these, although most of the human cathepsins K are having a similar or identical similarity, but in this study, I only considered human cathepsin K. But however, other cathepsins K are also an open gateway to have a study. To understand with this, I mostly had a focus on the orthosteric, means the active binding pocket groove, and what are the active residues, so that I can distinguish which can have some kind of difference between the falciparum, Plasmodium versus human cathepsins. So among this, there is total 4 subtypes, as it's a human cathepsin K, and here there is a striking differences which I encircled here. So mostly the main difference is in the selectivity side, H2 subside, Plasmodium falciparum have a acidic residue, while human cathepsins K have a neutral residues. So designing any molecule which having a polar kind of interaction have a much more susceptible to bind with falciparum, but not with human cathepsins. Although the binding affinity, the absolute binding free energies will have a higher frontier, means higher relativity, but however, this have to be validated through the biological studies. So these are some supporting rationale papers which has been reported. The things that I have been narrated so far, this has been mostly supported with these reported articles. These reported articles are also been studied by considering human cathepsins K, and these are the active conformer that I mostly probed of those 2 identified compounds, NM12, 115, along with all 3 proteins, and these are the RMSD, means the accurate docked conformer, which has been validated using binding pose metadynamics. And these are the few interactions. Mostly I am studying on the non-covalent interactions, mostly the hydrogen bonding interactions. So, and which has been validated through the classical MDS. Now, so, and this is mostly the principle of multidimensional PCA analysis of all the targets along with the two molecules, two inhibitors, NM12 and NM15. And this is the dynamics video, how the consistency of the all the molecules been studied here. So mostly this is showing the conformation of the active pocket. Here mostly the interactions has been hydrophobic because the active residues are surrounded with the neutral residues. So I have to optimize in a such a way that it can bind only with falciparum with a polar interaction, but not with human. So I try to curtail it down. I reduce the structural changes where I kept where I reduced the hydrophobic, means the spacing of chain group. Further, I had studied how these changes make affected positively along with these three proteins, means with three drug targets. So interestingly, surprisingly, I found that falciparum have a good affinity. Mostly in this study, I considered NBE is means non-bonded energy. Non-bonded energy is the cumulative of electrostatic plus Coulomb and van der Waals energies, and where Le is the ligand efficiency. Ligand efficiency means
Even your time is about to just complete it within 1 minute.
Okay, ma'am. Okay. So after the optimization, I try to find out the accurate conformer. So based on the structural diversification, I try to have optimized the spacer backbone and perform the lead optimization mostly at the subsite 2 that can inhibit. So mostly I try to occupy the polar side chains, which is forming a hydrogen bonding interaction with falciparum, but not with any human cathepsins. So in my molecule, there is also metabolic hotspot, which is susceptible to interact with multiple CYP450 isoform. I try to also minimize with that, so it won't undergo any biotransformation pathways. So in the conclusion, I found that two of the molecule NM12H and NM12G is mostly been highly potential compounds, which can be further designed for the analogs. These are the interactions of the final molecule, and this is the mostly the best molecular dynamics of the final leads, where I also confirmed with the control group. Although there is no clinical pipeline so far, I took the control group here as a co-crystal, which is a covalent ligand bounded A64 and K11017, which I have about more than 1.5 fold of affinity from the parent and the control group. This is mostly the assessment of the physicochemical, and I'm concluding, and now I'm open for the discussion.
Judges are requested to ask the question.
Jeevan, I have one question. You can hear me, right?
Yes, ma'am.
Yeah, you have done plenty of work and impressive work, quite impressive work. You said that in the active pocket site, there is a leucine in plasmodia, right? Leucine that you have highlighted, but in human, it is a glutamine.
Ma'am, just opposite. Leucine is in human cathepsin. Neutral.
Okay, yeah, vice versa, like let's say, but how it can be if you're going to disturb the active pocket by changing the amino acid, then there, because leucine is a highly hydrophobic molecule, and right, and possibly it cannot be replaced with the uncharged polar group. So it can disturb the configuration of the protein or that enzyme also, and then yeah, you're saying something.
Here I didn't perform any kind of single point mutations. This is the protein sequence alignment, which where the leucine is already been there as a wild type.
Okay, but not in the that organism, but it is in the human, you said, right?
Yes.
Okay, so that's what I'm asking. You're doing comparison, and you found there is a in the same active pocket in human there is a leucine, but there it is a glutamine. So, do you think these things when you are going to change it will not disturb the active pocket site or activity of the targets, and it will give the similar binding energy interaction?
No, first of all, I didn't perform any say any mutations, alteration of amino acid.
Yeah,
I
this is
took
bioinformatics you are doing to find the binding pattern, right? That's best binding one,
I didn't
interaction
found-
basically.
Yes, ma'am. I didn't found any kind of glitches or any distortion of the whole protein because I took the wild type of all proteins. I didn't perform any kind of mutations to confirm whether this mostly leucine is responsible for the osteoclast, mostly
Yes.
osteorelated. So, Plasmodium having a acidic at the selectivity subside where human cathepsins doesn't. So based on the wild type only, I distinguished and I didn't found any kind of glitches throughout the studies.
Okay.
Okay, Jeevan, I have one question.
Sure, sir.
You went for this drug discovery of the falciparum inhibitors, right? And in your study, I have seen your table in which there was the representation of Gibbs free energy that was minus 47 degrees Celsius. I can see there. But the lower Gibbs free energy is also there. So why you are pointing that NM molecule that you selected there? Can you go to the table? Can you see there? Yeah, this one. No, not this one. I think there was the 47 degrees Celsius that you have selected the NM molecule. Yeah, here you can see the minus 47, but I can see here also in case of NM12K, the Gibbs free energy is minus 42. So what was the reason to select this 47? We cannot go for this 42?
No, actually, here I studied the metabolic hotspot, where here in the last column, I here mentioned 3A4 CYP isoform and 2D6 having a much more interaction. Due to the biotransformation pathway, the metabolites of drug will make some metabolites changes. So I consider 47.96 because I didn't found any kind of hits prediction with any kind of CYP 450. So I considered this one, NM12H.
So you have seen the residues also here?
Yes, sir. And finally concluded. These are the potential residues and final binding post metadynamics.
Okay. So have you done any simulation or not?
Yes, sir. These are the simulations.
Yeah.
I collected all in a single
Which software
infographic.
you have used?
The whole study is performed on Desmond.
Okay. Thank you so much.
Oops.
Jeevan, unshare your screen now. The next presenter is Miss Deepali.
Hello.
Deepali, connect yourself.
Yeah, sure.
Deepali, turn on your camera.
Yeah, sure. The presentation is visible in my screen.
Yeah, presentation is visible.
Yeah, I have shared the slideshow. I think it's taking time in the screen.
Okay.
Okay. Good afternoon, everyone, and thank you for allowing me to present my work. Actually, it is a review work based on my PhD thesis. So, the topic of today's presentation is an overview of selected heavy metals, basically aluminum, arsenic, cadmium, lead, and mercury in Indian bovine milk. It can be a cow's milk, buffalo, goat, or camel milk, the possible sources, and their human health impacts. So basically, we all know what heavy metals are. They are basically defined as a metallic elements that have relatively high density compared to water, but with the assumption that heaviness and toxicity both are interrelated. So these metals can include either metalloids such as arsenic, cadmium, mercury, lead, and many others that are able to induce toxicity at very low level of exposure. So basically, when we talk about them in food intake is one of the way through which the contaminants they enter in the body. There are other various chemicals that may also enter with a variety of means. These chemicals can be substances like flavoring.
Sorry, Deepali, your slide is not in the slide share mode. Can you make it?
Yeah, sure, ma'am.
Thank you so much.
Are they moving now, ma'am?
Not yet. You have to press F5 or either way. The zoom, you see the zooming area, there is one umbrella-like things. You just press that.
Yeah, ma'am, I've just.
Yeah.
Okay. So these metals, they have chronic concerns.
Sorry
Is it
to
now?
interrupt you. Not yet. I think you have shared the slide, but not the screen. So I guess you need to do the, or you can move it. You just move. You have to click one by one then, okay?
Yeah. Is
Otherwise,
it
yeah.
visible now?
Yeah, now we are in third slide, but it is still in
Yes,
the
ma'am.
zoom
Yeah,
in.
ma'am. Actually, I'm sending it to slideshow, and then the slides are, I think, not visible to others. So can I continue this way?
Okay, continue.
Yeah, thank you, ma'am. So basically, these metals, they have a chronic concern globally as they have been considered as a hazardous contaminants found affecting the environment and causing toxicity basically in human and animal health. So basic main reason can be increase in urbanization, industrialization that contaminates the soil, water, and air, and basically that reaches the food and water intake. So these metals have relatively low atomic weight, as I said earlier, and they are basically toxic whenever available in excess amount, even taken for a longer period of time. So they can be contaminating vegetables, tubers, fruits, milk, basically water. That may attribute to multiple range of sources of human activities such as metal-based fertilizers or pesticides are used during farming or any industrial or domestic wastewater. Other than this can be food items that can be contaminated with metals by pollution or industrial exhaust or during transportation and marketing. Some amounts of metals can also be introduced in food items during processing methods. There are various metals like arsenic, cadmium, chromium, lead, mercury. They have been studied by different researchers in foods from around different areas of India and other countries also. And when humans get exposed to these metals for a longer period of time, their health negatively affects. So these metals are harmful to organs. Uh, First of all, I would like to introduce what can be the sources. So they can be agricultural sources or industrial sources, or there can be domestic sources, including inorganic or organic wastes, or maybe uses of batteries, biomass burning, e-waste that can lead to lead toxicity. And other foods, so other sources can be fossil fuels burning, landfills increasing the dump sites, or other pollution causing factors. Natural sources can be like volcanic eruptions, weathering of soils and rocks, and other. Basically, they transfer metals to animals from plants, soil, and water, and the transfer of metals to human body through the different food chain methods. What can be the health effects of these metals? They in overall they can cause reproductive or developmental effects, immunological disorders, liver diseases, cardiovascular diseases. Basically they affect the organs. There can be some signs or symptoms in the earlier stages like diarrhea, stomach pains. There can be vomiting cases, bone fractures. They can be damage to nervous systems, immune system, and in children's basically that there is seen the psychological disorders. They basically lead to high levels of lead can cause basically anemia, weakness, kidney. Cadmium, arsenic, lead, and mercury. They are considered as toxic carcinogenic elements. And when we talk about aluminium, it basically causes Alzheimer's diseases, Parkinson diseases, and other multiple sclerosis. Arsenic is basically famous for causing skin or eye related diseases, and they have also negative effects on different organs. So I have studied several Indian studies. Basically, I'm currently working on this review. Till date, I have found these researchers that are basically showing the contamination or the levels of uh, metals in milk. That can be a buffalo milk, a cow's milk, or goat milk, or they have maybe used different products, different branded milks. So in different studies, I found that in Maharashtra, basically, they have found buffalo milk that has been found a good level of lead. In Hyderabad, same buffalo milk has found a level of lead. In Tamil Nadu, also they studied in goat milk and arsenic, leadmium, and mercury. They four have been studied. In Vadodara also they have conducted analysis in milk and milk products and branded milk also. So while we compare, so branded milk product having a low contamination of or low levels of cadmium than the milk products. Same goes with Mumbai and other studies being conducted in different areas of India. But addition to this, my PhD thesis work was also in assessment of these five metals in different food groups and milk products and water. So through my study, I found that I have conducted the study among Jaipur and Bareilly. So in my study, basically aluminium was within the limit, but other metals, arsenic, cadmium, lead, and mercury, they were exceeding the limits as being given by the recommended or the permissible limits given by different, organizations. So basically, I am currently working on the review process. Uh, After this, I can conclude that metals may contaminate animal feed through different instruments, or they can be different machinery or processing techniques, or during the distribution of milk that can be contaminated. Processed milk is being reported to have higher concentration of heavy metals as compared to the raw milk. And I've also found in studies that cadmium is found to be more than lead in the raw milk, and also that buffalo milk was, however, having more contamination or more levels of metals than the cow's milk. And what was the basic reason? I'm also studying right now on that theme. And heavy metals, they can enter through milk by you, maybe the animal feed is contaminated by the roots of irrigation, or that using polluted canal or sewage water in the animal feed, or the application of pesticides and fungicides are used in the areas or grasses or the crops on which these animals are grazing. And the content of raw milk is found a higher heavy metal concentration in milk from areas in proximity to industrial zones with significant vehicular traffic, while lower heavy metal concentration in the milk sample from rural areas. High level metals and levels in milk samples collected from shops and processed milk as compared to milk samples collected directly from the farms were also studied. And in the present review, a number of reports have been I have collected, and recently I'm also collecting some other reports also. Among these reports, I have found that TB and CD in the milk samples have shown elevated levels than the mercury and arsenic. So I can add upon to this that treatment of industrial wastewater by absorption of heavy metals using different methods to, remove the
Dipali,
different
so
can
your
lead to
slide
this one.
is not moving. Just go back to the slide. Okay, the exact slide what you are delivering.
Sure, ma'am. Oh, I can share with you.
Okay, just conclude in a simple manner. No need to express.
Okay, ma'am. So lastly, I was just concluding that using different, technological methods or industrial waste removal methods in basically metal adsorption techniques can be used to remove the contamination from the water bodies or the feed that is being feeded to the animals. And there are different most advanced treatment technologies such as nanofiltration, reverse osmosis, and ion exchange also. So basically, changes in milk manufacturing processes can also help to reduce the contamination of milk by heavy metals. Water used for drinking of animal and fodder of milking animals should be regularly monitored so that they can be evaluated, and recommendations can be taken to remove or to less the contamination. Precautions should be taken to avoid the metal contamination during handling, processing, and storage of milk and milk products. And only food grade materials should be used during these steps. Further, I can add that consumption of milk in some areas nearly are free of risk, but bioaccumulation of these metals through the food chain or through milk can lead to, health concerns. Special attention should be given to these metals as they are present in concentration greater than the acceptable daily intakes. So it may be difficult to reduce them at an acceptable level. So pre methods or precautions should be taken by different agencies, or some policy making can be done to assess these concentration and work accordingly. Um, Thank you from my side.
Judges, you are requested to ask the question.
Thank you, Dipali. I would like to ask one thing, whether you have compared that which metal is accumulated more in the milk, in bovine milk, because we
Yes,
have
sir.
talked various type of minerals.
Yes, sir. I have actually worked in these five metals in on my own study, and I have found that basically among the five metals, we can say that cadmium and lead, they were found more as compared to other metals. And as I have studied the other studies also, same goes with cadmium and lead. So these two metals have been found contaminating more the milk and other food products also.
Any metal with respect to specific type of bovine milk, means suppose cadmium is more found in goat milk or some
Yeah,
other.
sir. Yes, sir. Yes, sir. Cadmium has been found more contaminated in more level in mainly in buffalo milk.
Buffalo milk, okay.
As these studies they have told in their studies, but we are also planning to have a study in our area so that we can assess that which among these metals are being contaminated in raw milk. So either in which type of animal, either buffalo, goat, any cow or camels.
So whether this type of accumulation is geographical based, means suppose if any cadmium rich area or arsenic rich area is there, so we can find the concentration is more
Yes,
in
sir.
comparison to others.
Yeah, we can compare with the geographical area, as we can take the sample from the animal and then we can take the sample from where maybe the exposure. So we can then compare. Even if, for example, if the area is an industrial area and there are more of the industries that are working in, maybe we can say paintings or painters or lead batteries, so we can compare that maybe the lead is increasing in those grazing area animals or not. So we can have a comparison that the geographical reason is being affecting the contamination or concentration or not.
And
So
secondly,
my
whether
research,
we have found this particular type of heavy metals in the packaged milk also.
yes, sir. I haven't conducted the studies, but researchers have said that the branded milk products are having a level, but not all are having a contamination level. Means they are not exceeding. They all are not exceeding the permissible limits. But when we compare it with the raw milk is having more levels, contamination levels.
And can we compare it with the toxicity level? Means because some of the metals which are responsible for a neurotoxicity or some are responsible for the kidney diseases or like this. So can we relate
Yes,
with
sir.
these type of heavy metals with the diseases?
Yes, sir. Then we need to have to conduct, blood samples also, so that we can assess that particular metal is present or not. If the person is having any kidney related or any specific disease related, and then we can compare the different types of method or exposure routes, including food, water, fumes, or any maybe they have any occupational thing that maybe they are working in any industry in which they are in direct contact with the toxicity causing work.
Okay.
So then we can compare.
Okay. Thank you, Deepali. Thank you.
Thank you.
Okay, the remaining students name I'm calling, those who is still not present their work: Dr. Somali Sanyal, Ms. Brinda Devi, Ms. Archana Agrawal, Ms. Patel Dhruvi, Mr. Himanshu Sharma. These candidates are there. Please raise your hand. Otherwise, we will close the session.
Yesterday, I have received a message from Dhruvi that there is some change in name. Actually, she is Miss Dhruvi Patel, but it has mentioned Mr. So she requested to change her name to Miss Dhruvi Patel, and she is willing to come for the presentation, but I don't know why she is not here.
Sir, she is in the attendees list and I have promoted her as a panelist. But
Okay.
maybe I don't think so. She's Dhruvi. Please raise your hand. Okay, now I'm going to conclude the session. So I would like to request our coordinator, our director, sir, Dr. Professor Vidya Duveri, sir, to conclude the session, sir.
Thank you, Dr. Asha, and thank you very much. Dr. Sushma, thank you for coordinating the session along with me.
Thank you so much, sir.
Shushma ma'am, please turn on your camera also, so we'll take a group photograph.
Yes.
Sure, ma'am, sure.
And thank you, participants. Thank you, presenters. Thank you very much. In the valedictory session, we will announce the name of the best paper presentation in every session. So be there.
Sure, sir.
Thank you.
Hope to see you offline sometimes.
Yeah, sure.
Yeah. Thank you so much, sir. Thank you so much, ma'am.
Thank you so much, ma'am, for
Thank you, ma'am.
connecting and delivering the very informative lecture, ma'am. So we will
Thank
meet on the valedictory section, ma'am.
Sure, ma'am, sure. Thank you so much.
Thank
Thank you
you.
once again, Amity
Thank you.
University, Gwalior.
Thank you.
Thank you, sir.
ಹೇಳಿ ಹೇಳಿ
ಸರ್ ಇದು ಇವರು ದೀಪಾ ಅವರು ಸುಗ್ನಾ
ನೋಡಿದೆ
ಒಂದು ಮೇಲ್ ಕಳಿಸಿದಿಲ್ಲ
Very good afternoon, sir. Yes, yes, sir. एक minute. Hello.
आवाज आपकी इको आ रही है। Welcome, Dr. Gowda. This is Dr. Vinay Dwivedi, Director AIB. Welcome to the validity function, sir. Hello, sir. Oh, okay. I have done that. Okay, I did that. Okay, okay. नहीं मैं अभी 4:30
A very good afternoon to one and all present here. I, Dr. Neha Sharma, welcome you all in valedictory session of Third International Conference on Recent Advances in Biotechnology and Nanotechnology. We have been listening to eminent speakers with their ideas in the emerging areas of life sciences since yesterday and today's morning. All these valuable insights gave us a new perspective that one should adopt while discussing these issues. Dear all, we have with us Dr. Channagoda, MBSc Vice President, Technical GS Biotech Private Limited, Mysore, Karnataka, as the chief guest of the valedictory session, and Director AIB Professor Dr. Vinay Dwivedi. May I now request Professor Dr. Vinay Dwivedi, sir, to kindly address the session.
Good afternoon, everyone. The chief guest of validity session, Dr. Chenna Gowda, VP GS Biotech, distinguished speakers, researchers, and participants. It is with great pleasure and honor that I stand before you today to deliver the opening remark for the valedictory session of the Third International Conference on Biotechnology and Nanotechnology, BioNano 2024. This event has been a testament to the remarkable progress and collaboration within the field of biotechnology and nanotechnology, bringing together brilliant minds from across the globe to share insights, discuss advancements, and forge new pathways for the culture. As we gather here, we find ourselves at the intersection of two groundbreaking scientific domains, biotechnology and nanobiotechnology. These disciplines, though distinct, have converged in recent years, giving rise to unprecedented opportunities and innovations. The amalgamation of biological sciences and nanoscale technologies has led to the transformative breakthrough with profound implications for medicine, agriculture, environment, sustainability, and various other sectors. Over the last 2 days, we have witnessed a plethora of presentations, discussions, and poster sessions that have delved into the depth of biotechnological and nanotechnological research. The sheer diversity and depth of the topics covered are a testament to the dynamism and rapid evolution of these scientific fields. Researchers from academia, industry, and government institutions have come together to share their findings, discuss challenges, and pave the way for collaborative endeavors that promise to shape the future of science and technology. One of the key themes that have emerged from this conference is the role of biotechnology and nanotechnology in addressing global challenges, from healthcare to energy, from agriculture to environment. These disciplines offer innovative solutions that have the potential to revolutionize the way we live and interact with the world around us. The research presented here has showcased the power of biotechnology and nanotechnology to enhance the quality of life, improve resource efficiency, and contributes to sustainable development. In the realm of healthcare, we have seen remarkable strides in the development of nanomedicines, targeted drug delivery systems, and diagnostic tools that promise to revolutionize the diagnosis and treatment of diseases. The ability to manipulate biological systems at the nanoscale has opened new frontiers in personalized medicine, allowing the tailored and precise interventions that minimize the side effects and optimize therapeutic outcomes. The potential for nanotechnology to reshape the landscape of healthcare is immense, and the research shared at this conference is a testament to the dedication and ingenuity of the scientific community. In agriculture, the biotechnological advances has played a pivotal role in addressing food security challenges, from genetically modified crops with enhanced resistance to pests and diseases to nanoscale sensors for precision agriculture. The intersection of biotechnology and nanotechnology hold immense promise for sustainable and resilient agricultures. As the global population continues to grow, the innovations discussed in this conference provide hope for a future where we can meet the growing demand of food while minimizing the environmental impact of agriculture practices. Environmental sustainability has been another focal point of this conference, with discussions on the role of nanotechnology in water purification, waste management, and pollution control. The ability of nanomaterials to efficiently capture and remediate pollutants opens new avenues for creating a cleaner and healthier planet. Biotechnological approaches, such as the use of microorganisms for bioremediation, complement these efforts and offer sustainable solutions for environmental conservation. The insights shared at this conference underscore the importance of harnessing the power of biotechnology and nanotechnology to address the urgent environmental changes facing our world. Collaborations has been a recurring theme throughout this conference, emphasizing the interdisciplinary nature of research in biotechnology and nanotechnology. The synergy between biology, chemistry, physics, and engineering has led to the groundbreaking discoveries and innovations that would not have been possible within the confines of a single discipline. The collaborative spirit showcased here is a testament to the collaborative determination to overcome complex challenges and push the boundaries of scientific knowledge. As we reflect on the achievement and discussion to the last 2 days, it is crucial to recognize the role of education and outreach in fostering the next generation of scientists and innovators. The insights and experiences shared in the conference provide a wealth of knowledge that must be disseminated to a broader audience. By nurturing the curiosity and passion of young minds, we can ensure a continuous flow of ideas and advancements that will shape the future of biotechnology and nanotechnology. So thank you all for your contributions, and I wish your continued success in your endeavors. Thank you very much.
Thank you so much, sir, for addressing us. Now, it is my moral duty and my pleasure to introduce our chief guest. Dr. Chennai Oda started his career as a field veterinarian in the year 1992 and took up animal nutrition to support his passion for improving animal health and performance. Having put in vast experience in the area of animal health, ethics, clinicals, animal feed additives, etc., he has been assisting farmers, animal owners in the South Karnataka region of India. He is also involved in quality control of animal feed and feed ingredients to feed manufacturers and poultry farmers. He has traveled across South Asia, Middle East, and East Africa in more than 10 countries. He holds a bachelor degree in veterinarian science and a master degree in poultry science from the University of Agriculture Science, Bangalore, India. He was winner of 3 gold medals for highest merit, and he has published 2 research articles, many short communications, and popular articles. He was appointed as assistant professor to Hassan Veterinary College in the year 2013, but he preferred to step back in the Department of Animal Husbandry and Veterinary Services. He was a resource person in the department and worked in control and containment of bird flu INAPS registration of animal feed mills, etc. He has also worked as a research associate at University of Kentucky, USA for a brief period, presently working as GS Biotech Private Limited, Mysore as Vice President since 2019. Please, sir, kindly address the session.
Thank you very much, madam. That is a very nice introduction of mine. Good afternoon, everybody, and namaste. Honorable Pro Chancellor of the University, speakers, delegates, participants, guests, ladies and gentlemen. At the outset, I would like to thank the organizing committee and the university for giving me this opportunity to address as a chief guest. And I'm very happy here, delighted to be among with you on this occasion of International Conference on Nanobiotechnology. Well, basically, I'm not a scientist. I'm an animal doctor, but I'm working in the animal feed biotechnology field from last couple of years. But no doubt, I'm a strong believer of science and practicing science, and I'm end user of your technology. So you can depend on me for your feedback and the field evaluation, definitely. Right? So in the last few decades, the biotechnologically derived molecules such as nanoparticles have found their wider applications in various areas, including agriculture. Or to me, it is animal agriculture, livestock production. And then food safety issues. To me, again, it is animal feed safety issues, like encapsulated probiotics and nanoparticles of trace minerals, mineral supplements, some of the biotechnologically derived enzymes like phytase in animal nutrition. And that apart, the pharma industry for efficient and targeted delivery of these drugs and vaccines. And then cosmetic industry, because these nanoparticles, as I understood, they are biocompatible. They are highly compatible with the biological system. They are biodegradable, so that eco-friendly, and their inclusion rate is very less, and so that it can reduce the cost of inputs, and they are non-toxic also. So considering these advantages, nanoparticles have drawn wider attention from various fields, especially in the precise and targeted delivery of drugs, various drugs, for both for human medicine and also for veterinary medicine in our case. And also better presentation of the antigens, viral antigens especially, or bacterial antigens to the immune system, so that we can elucidate a better immune response from the animals or human beings. So with these kind of things, not only improve our fundamental knowledge on biotechnological derived nanoparticles, but also guide us in designing various potent therapies in the future. Especially, we are facing a tough challenge of antibiotic resistant pathogens in the field of animal agriculture. So we usually fight with the medicos and the veterinarians for the where is the drug resistance coming from various parts of the practicing field, both from human medicine and also from the veterinary medicine. Now, these molecules definitely will help us in designing a kind of potent therapies, especially combination therapies. Maybe a nanoparticle can be combined with one of the antimicrobial agent, may be delivered better to address the antimicrobial resistant pathogenic infections, right? So in this, again, we should also think of contemporary technologies such as artificial insemination, artificial intelligence. So artificial intelligence can be adopted along with the nanoparticles technology to give a better delivery again and to give better efficiency for the systems. Right, sir? So the roadmap is not so easy again. I mean, the road ahead is again a tough challenge. It is not so easy. There are challenges too, because since these are biologically active molecules, we have to evaluate their safety, whether they are highly safe, how compatible, and is there any side effects, or is there any development of resistance? Because end of the day, it is a biological molecule. So we need to evaluate more and more, and research studies are required to evaluate their mutagenicity, whether that molecule is inducing any mutations. Genotoxicity, is that molecule is causing any genotoxicity, and their interactions with other biological molecules, and how the resistance mechanism is working within the system with these molecules. So more research is required in this area. So I really thank the organizers once again for giving this platform, because this platform has given opportunity for all of us to share our views and experience and the knowledge. Finally, I would like to thank all the participants, delegates, guests, and the eminent speakers, experts, for sharing their knowledge, views, and experiences in making this conference a grand success. I hope the take-home messages from this conference will definitely impact all of us and will help in building our nation and in turn the mankind. Thank you very much.
Thank you so much, sir, for addressing us with your knowledge and experience. Thank you once again, sir. Now moving to session for prize distribution for Best Presentation Awards. For the same, I request two respected chief guests, Dr. Sushma Chauhan and Dr. Pankaj Rai, one of our speakers, and Director AIB, Professor Dr. Vinay Dwivedi. Please appreciate winners. For the same, I request Director AIB, Professor Dr. Vinay Dwivedi, kindly announce the result. Please, sir.
Thank you, ma'am. Thank you very much. So now I'm going to announce the name who has bagged the best presentation prizes in different sessions. So I will start one by one. So first presentation, Ms. Neha Thakur from Graphic Era University, Dehradun, Uttarakhand has bagged best presentation prize. Second, Mr. Prateek Singh from Amity Institute of Biotechnology, Amity Institute of Lucknow campus has bagged best presentation prize for his research paper titled Characterization of PGPR on Phosphate Solubilization. Ms. Raja Kumari K from Wells Institute of Science, Technology and Advanced Studies, Chennai has bagged best presentation prize for her research paper titled Molecular Simulation Studies and Systems Biological Approach to Identify the Target-Led Compounds for Enzyme DNA Gyrase. Mr. Krish Thakkar from Department of Biotechnology, Parul University, Parul Institute of Technology, Parul University, Vadodara has bagged best presentation prize for his research paper titled Effect of Plant-Based Silver Nanoparticles on Seed Priming in Salt-Stress Indian Mustard Brassica juncea seedlings. Mr. Moksh Rajpal from Chandigarh University has bagged best presentation prize for his research paper titled Innovative Process of Sustainable Beer Production from Fruit and Vegetable Waste, a holistic approach towards process optimization and cost efficiency. Ms. Anushri S. Nair from Sri Satya Sai College of Women, Bhopal has bagged best presentation prize for her research paper titled Biodegradation of Toxic Chemical Pesticides by Inoculum of Nitrogen Fixing Bacteria and its Synergistic Effect on Soil Fertility. Ms. Neha Srivastav from Amity Institute of Biotechnology, Amity University, Madhya Pradesh, Gwalior has bagged best presentation prize for research paper entitled Exploring Microalgae for Bioabsorption of Toxic Nanoparticles. Ms. Roshni Lalwani from CVM University, Vallabh Vidyanagar, Gujarat has bagged Best Presentation Prize for her research paper titled Decoloration of Synthetic Dyes Using Copper Oxide and Iron Oxide Nanoparticles. Ms. Megha Chaube, Ms. Kiran Tomar, and Ms. Anjali Devi M. from Amity Institute of Biotechnology, Amity University, Madhya Pradesh, Gwalior has bagged Best Presentation Prize for their research paper titled Fueling Sustainability, Transforming Waste Cooking Oil into Biodiesel. Mr. Harsh Pratap Singh Kushwaha from Amity Institute of Biotechnology, Amity University, Madhya Pradesh, Gwalior has bagged Best Presentation Prize for his research paper titled Mycelium Bricks, a Sustainable Paradigm Shift in Construction Material. Ms. Vargobi Mukherjee from Amity Institute of Biotechnology, Amity University, Chhattisgarh, Raipur has bagged Best Presentation Prize for a research paper titled Exopolysaccharide EPS Production by Bacteria to Remove Heavy Metals from Environment. Mr. Jeevan Patra from School of Health Sciences and Technology, University of Petroleum and Energy Studies, Uttarakhand, has bagged Best Presentation Prize for his research paper titled Structural Binding Modes and Optimization of Dual Falcipain Inhibitors Using Computational Approaches. Mr. Aditya Raj from Central Ayurveda Research Institute, New Delhi, has bagged Best Presentation Prize for his research paper titled Molecular Detection of Certain Pathogens from the Feces of Bats. And the last, Mr. Surya Prakash from Maharshi Markandeya University, Mullana, has bagged Best Presentation Prize for his research paper titled Transformative Diagnostics for Infective Keratitis, a Molecular Approach. Thank you, and best wishes for the Best Presentation Prizes.
Thank you so much, sir, and congratulations to all winners. May I now request Professor Dr. Vikas Srivastava, co-convener of the conference, to kindly give concluding remarks. Please, sir.
Thank you, Dr. Neha. And once again, thank you very much to all the dignitaries. So, like, the journey of 2 days of this conference is about to finish. So I would like to conclude all the sessions and whatever activities has been carried out during the conference. Amity Institute of Biotechnology organized the 2 days third national international conference on recent advances in biotechnology and nanobiotechnology in hybrid mode. The inauguration of the conference was in the physical mode, and all the participants were basically joined with in the online mode. During the inauguration, Dr. P.S. Besen, former Vice Chancellor, Jiva University, chief guest of the event, was present. Along with that, Dr. A.K. Meena, Institute in charge, CCRAS, Regional Ayurveda Research Center, Gwalior. Honorable Pro-Chancellor, Lieutenant General V.K. Sharma, AVSM, officiating Vice Chancellor, Professor Dr. Anil Vasisht, Pro-Vice Chancellor, Professor Dr. M.P. Kaushik, and Professor Dr. Vinay Dwivedi, Director AIB, was present. The inauguration was done with the garlanding of Ma Saraswati and Saraswati Vandana. In the inaugural session, first of all, Honorable Pro-Chancellor Lieutenant General V.K. Sharma gave the inaugural address. He elaborated on the application of nanotechnology for human welfare and mankind. After that, the chief guest of the conference, Professor Dr. P.S. Besen, addressed the audience and emphasized the use of recent technologies like artificial intelligence, machine learning in the field of biotechnology. He also discussed the emergence of biotechnology in the country. Later on, the abstract book of the international conference was released by the dignitaries. After that, the first keynote lecture was delivered by Dr. Mukesh Dubey, Associate Professor, Swedish University of Agriculture, Uppsala, Sweden, on the topic of RNAi technology in plant disease management, followed by second keynote address by Dr. S.I. Alam, Scientist F, DRDE Gwalior, on the topic of proteomics as a new experiments. He emphasized the mass spectroscopy based proteomic studies. After that, there were 3 technical sessions in day 1, in which total 15 speakers from reputed international and national academic institutions delivered their invited talk, including keynote address. In first session was basically molecular biology and plant biotechnology. And the chairman of the session was Dr. Pranav Kumar Mandal, Principal Scientist, National Institute of Plant Biotechnology, LBS Center, Pusa Campus, New Delhi. The session was chaired by Dr. P.K. Mandal and Dr. Vinay Dwivedi. The second invited lecture in this session was given by the Dr. Siddharth Tiwari, Scientist F, National Agri-Food Biotechnology Institute, Chandigarh, on the topic of plant application in crop biofortification. Followed by next invited talk by Dr. Tanushree Kaul, Group Leader, Nutritional Improvement of Crop, International Center for Genetic Engineering and Biotechnology, New Delhi. The another talk was given by Professor Dr. Rajesh Patel, Department of Biosciences, Veer Narmada University, Gujarat. The topic of the talk was Unveiling the Secret of Nature, a metagenomic and metabolomic exploration of natural farming inputs for sustainable agriculture practice. And the last talk of the session was delivered by Dr. Bhaskar from Savita University, Chennai, India, on the topic of glucosinolates, a sulfur-based compound, their synthesis and application perspectives. Later on, the second technical session was based on the nanotechnology and biosensors. This session was chaired by Dr. Mukesh Gupta, Director ICMR, NAR, FBR, Genome Valley, Hyderabad, Telangana, and co-chaired by Professor Dr. Vinay Dwivedi, Director AIB AUMP. The Dr. Mukesh Gupta delivered an invited talk on the topic of ketosine-based nanofibrous PEC for rapid homeostasis. Afterward, the another invited talk was delivered by Dr. Brajesh Kumar, Head, Tata College, Kollam University, Chaibasa, on the topic of green synthesis of metallic nanoparticle and environment remediation. After that, the Dr. Muthu Trivengandam, Department of Crop Science, College of Sanghou Life Sciences, Konku University, Seoul, South Korea, on the topic of exploring the infinite frontiers of nanobiotechnology innovation, challenges, and application. With this, the technical session 2 was over. Then, in the late evening, third technical session was based on the secondary metabolites and metabolic engineering, which was chaired by Dr. Anurag Sharma, Associate Professor, Department of Environmental Health and Toxicology at Nitte University Center for Science Education Research, Nitte, Mangalore, along with Dr. Vinay Dwivedi, Director AIB. Dr. Sharma chaired the session. He delivered a talk on benzene and hematopoietic response, drawing insight from Drosophila. After his lecture, Dr. Praveen Avasthi, postdoctoral research fellow, Department of Plant Molecular Physiology, Institute of Bioorganic Chemistry, Polish Academy of Science, Poznań, Poland, gave the lecture on the topic of CRISPR toolbox for Hope Metabolic Engineering. Very first day, the 3 parallel session was also conducted on the Google Meet, where the different chairpersons and co-chairs were available, and total 60 presentations were concluded. Then second day of the conference, the first session, the technical session 4 was started The theme of the technical session was Animal and Medical Biotechnology and Ethics in Research. This session was chaired by Dr. Naresh C. Bal, Associate Professor, KIIT University Bhubaneswar, and co-chaired by Dr. Vinay Dwivedi, Director AIB, AUMP Madhya Pradesh. Dr. Naresh delivered an invited talk on the topic of targeting skeletal muscle-based NST towards countering metabolic diseases, identifying mechanism and molecule. And he pointed out some of the important skeletal muscles role towards metabolic disease management. Second invited talk was given by the Dr. Atul Sharma, Department of Critical Care and Respiratory Care Division of Anesthesia, Critical Care, Penn Medicine, University of Texas. He discussed about the mechanism of neutrophil extracellular trap, followed by invited talk by the Dr. Sai Maji, Health Scientist Administrator at the National Institute of Health, Rockville, Maryland, USA. The next invited speaker of the session was Dr. Akrup Banerjee, Cold Spring Harbor University Laboratory, USA. He delivered the talk on the how circuits of interacting neurons give rise to natural, adaptive, and behaviors. Discussed about the crosstalk of neural circuits, and the lecture was followed by the last invited speaker of the session, Dr. Anuj Sharma, Health Science Administrator, U.S. Department of Health and Health Services, Washington, D.C. He delivered about the implications of misconduct in scientific research on science and researchers, a perspective from the Office of Research Integrity. In total, there were 5 invited lectures were delivered during this session, and session was concluded with the panel discussion. After that, the another technical session, which is technical session 5, was based on the genetic engineering and bioinformatics. It was started by the Dr. Brijesh Barse, Director MedTech Life Science, Food and Agriculture, New Delhi, on the topic of importance of IPR in entrepreneurship and business development, specifically in biotechnology field. The session was chaired by Dr. Barse and Dr. Vinay Dwivedi. In this session, the second invited talk was given by the Dr. Pankaj Rai, Professor and Head, Department of Biotechnology, Invertis University, Bareilly. He discussed about the optimization of concentration of metal ions in spent medium for phyto biohydrogen production. Followed by the invited talk was given by the Dr. Sushma Chauhan, visiting scientist at Chemical and Biochemical Engineering, Dongguk University, Seoul, South Korea. And she discussed about the rings of innovation, circular proteins in contemporary biotechnology. The session was concluded with the panel discussion. That along with that, the total 4 parallel session was also conducted on the Google Meet, where somewhere 92 oral presentations were given. So now we are at the valedictory. This was the whole about the 2 days activities which was carried out during the conference. Thank you very much.
Thank you, sir, for concluding the sessions. May I now request Professor Dr. Raghvendra Saxena, Organizing Secretary of this conference, to give vote of thanks. Please, sir. Sir, please unmute yourself.
Good evening, ladies and gentlemen. It is indeed a great pleasure and my privilege to have been asked to propose a vote of thanks on this occasion. I, on behalf of MIT Institute of Biotechnology, MIT University, Madhya Pradesh, extend a very heartfelt gratitude to our Honorable Pro-Chancellor, Lieutenant General V.K. Sharma, AVSM retired, for support and providing encouragement at every point of time in the organization of this great event. My sincere gratitude to Honorable Officiating Vice Chancellor of Professor Dr. Anil Vasist and Pro Vice Chancellor Research, Professor Dr. M.P. Kaushik, AUMP, for unconditional support and encouragement. They helped us in obtaining the necessary administrative approval for organizing the event and supported in all possible manner unconditionally to make conference success. My special thanks to the chief guest of international conference, Professor Dr. P.S. Besen, former Vice Chancellor, Jiwari University, Gwalior, and guest of honor, Dr. A.K. Meena, Institute in charge, CCRAS, Regional Ayurveda Research Institute, Gwalior, for their gracious presence in the inaugural session of the conference. At this junction, I would like to extend a sincere thanks to our keynote speakers, Dr. Mukesh Dubey and Dr. S.I. Alam, for their deliberations to set the perfect tone of the conference. I also take this opportunity to extend our most sincere thanks to all our learned guest speakers who have unconditionally accepted our invitation to make the conference success and vibrant. I must mention our deep sense of appreciation and thanks to chief guest of today's valedictory session of this international conference, Dr. Channe Goda, MVSC, who is a vice president of ZES Biotech Private Limited, Mysuru, Karnataka, for accepting the invitation to grace this occasion. Thank you very much, sir. I extended my gratitude to all the chairpersons and co-chairpersons of various technical and parallel sessions occurred during the entire duration of the conference. Furthermore, I would like to thank all the delegates, participants, and students for their presence and participations to make this conference a success. I'm also very much thankful to the convener of this conference, Professor Dr. Vinay Dwivedi, Director AIB, AUMP, for constant guidance and support to shape this conference the way it has come out. I must take this opportunity to thank the entire conference organizing committee for their day-night untiring effort to make this conference success, which includes Professor Dr. Vikas Srivastav, Professor Dr. Raghavendra Kumar Mishra, Dr. Anurag Jyoti, Dr. Neha Sharma, Dr. Manish Kumar, Dr. Asha Singh, Dr. Raman Singh, Dr. Suren Singh Parihar, and Ms. Kirti Srivastav. I'm also very much thankful to Director Administration, AUMP, Colonel Sethi, and Brigadier Gangele for logistic support, and of course, the Team ASCO for media coverage, making the event entirely covered. I wish to express my thanks to IT team also of Amity University Noida and Amity University Madhya Pradesh for technical support, making the conference seamless going. In the last but not the least, I am extending thanks to all those whom I could not mention here for their support during the conference. And once again, we thank you for being with us this evening. It's been a great pleasure. Thank you. Thank you very much.
Thank you, sir. And thank you to all for their presence in valedictory session. May I request to all please open your camera for group photograph. Once again, thank you to all.
Thank you very much.
Thank you very much to all the invited speakers, all the chief guests. Thank you very much.
Thank you, sir.
We
Thank
look
you very
forward
much.
for the association, and definitely we would expect you in the next program wherever we will organize, and we need your supports and blessings.
So shall I close the session?
Yeah.
Thank you, sir.
Thank you.
Yes, Arvind, you can close the session.
Thank you all.